Hello everyone. Am I live? Am I back? Can you see? Can you hear? So can we start the class? Kindly confirm if I'm clearly visible, audible. Wait a minute. Okay. Thank you. Thank you for confirming. So let everyone join and we will finish bacteriology quickly. Okay. Let everyone join. So till now, what is the feedback? The bacteriology is clear. You don't have any doubt? We have completed many bacteria till now. So we have completed all gram-positive cocci, all gram-negative cocci. We have started gram-positive bacilli. And in gram-positive bacilli, we are continuing with the Clostridia. So we will quickly, in the next maybe 40 minutes or 1 hour, we will finish gram-positive bacilli will be there. And I'm going to take the gram-negative bacilli. Okay. Yes, in the 6th page, all the mnemonics are there. So can we start? Can we continue? Can we continue? So let everyone join next few minutes. Okay. So I will be starting with the Clostridium. So I have already started Clostridium. I will start from the beginning only. So I am teaching you gram-positive bacilli. In the gram-positive bacilli, we have already covered two big bacteria. We have already covered Bacillus and diphtheria, and now I am starting with the Clostridium. So I already told you how to do the classification. The classification of the Clostridium is based on RCM, whether they are proteolytic or saccharolytic or both or none. So this is the classification you have to learn, the classification of the Clostridium. Now there are many Clostridium, but don't worry, we don't study all. We will study only 4 of them. Just wait a while. Some technical glitch is going on, I guess. Wait a while. Am I clear? Am I clearly visible, audible? Okay. Am I clearly visible, audible? Can I continue? Can someone respond? Am I clearly visible, audible? Can I continue? Okay. Thank you. Thank you for responding. So there are many Clostridia, but we don't have to study all the Clostridia. We have to study only 4 Clostridia. The 4 Clostridia which are in our syllabus, these are the 4 Clostridia. So we have studied the morphology of them. So the 4 Clostridia, please learn in the sequence only, because in this sequence only I am going to continue. So the first is the Clostridium perfringens. The second is the Clostridium tetani. The third is the Clostridium botulinum. And the last is the Clostridium difficile. So these are the 4 type of Clostridium which are there in our syllabus. The Clostridium perfringens is also known as Clostridium wilchi. Please learn the other name. Now, I told you all the Clostridia are non-capsulated. They don't have the capsule except perfringens, which is the gaddar, going against of its family. All the Clostridia are motile, but this one is non-motile, going again against the family. So please learn in 2 ways it is going against the family. So it's a unique bacteria. It is going against its family in 2 ways. All are non-capsulated, but it is capsulated. All are motile, but it is non-motile. Please learn the 2 ways. But spore ke मामले में, in terms of spore, everyone have the spore. There is no exception. It is not any exception. So please learn that. So one by one, I'm going to teach you the 4 Clostridia. Let me start with the first one with full energy Clostridium perfringens. So let's start Clostridium perfringens. Okay. So let's start with the first bacteria, Clostridium perfringens. Can I start? So in Clostridium perfringens, this is the culture media. So I will tell 4 culture media, charon pe question hai. The first culture media is blood agar. Now in blood agar, I told you there are 3 possibilities. Some bacteria show alpha hemolysis, some show beta, some show gamma. Currently, I am teaching you Clostridium perfringens. It's a unique bacteria because it shows alpha and beta both. Yes, it is the only bacteria which show double hemolysis. So you can see here in the diagram also, you can see. So this is a colony. This is the colony of the bacteria around which I can first see the alpha, around which I can see the beta. So there is a colony at the center followed by alpha followed by beta. It's known as double hemolysis, target hemolysis. Very important PYQ. Double hemolysis, it's looking like a bull's eye. It is known as target hemolysis. So double hemolysis, target hemolysis, it is seen in Clostridium perfringens. Please learn on blood agar. Only bacteria which show double hemolysis, alpha and beta both. On RCM, Robertson Cook meat media, it forms pink color meat pieces. That is, it is saccharolytic. It is not proteolytic. I told you, the saccharolytic form pink color. On egg yolk. You know what is egg yolk? Egg yolk media. So let's take a egg yolk media. I'm growing the colonies of Clostridium perfringens. So these all are the colonies of Clostridium perfringens. Clostridium perfringens grow well on the egg yolk. So this is all egg yolk. Clostridium perfringens grow well on the egg yolk. You can see it is growing very well on the egg yolk. It is secreted toxin. The Clostridium perfringens have 12 type of toxin. The most important is alpha. The most important is alpha. है ना 12 होती हैं लेकिन हमें 12 नहीं पढ़नी. Most important is alpha. So alpha toxin ये secrete करेगा हर तरफ. It will secrete alpha toxin everywhere. है ना alpha toxin. The alpha toxin will create a zone of opalescence. The zone of opalescence, like blood agar पे होती है ना zone of hemolysis. Here the zone of opalescence. This is known as Niegler reaction. Niegler reaction. So it is Niegler reaction positive. Can you see the colonies? Around the colony, I want you to appreciate the zone of opalescence. This is zone of opalescence. This is known as Niegler reaction. And lastly, it is reverse CAM positive. What is reverse CAM? What I told you, CAM. CAM pe aata hai? Let's take a blood agar. On the blood agar, let's take a bacteria which is showing like Staph aureus. And perpendicular to this, let's take a bacteria like Clostridium perfringens. So here you will see the hemolysis. The hemolysis is more on this axis as compared to this axis. So this is reverse CAM. It is also a butterfly, but it is a reverse butterfly. The hemolysis is more on this axis as compared to this. Got my point? Please learn 3 bacteria. CAM के बारे में 3 चीजें याद रख लो. There are 2 bacteria which are CAM positive. मैंने दोनों पढ़ा दिए. You will tell which 2 bacteria are CAM positive. And there is, I'm sorry, मैंने एक ही पढ़ा है. And there is only 1 bacteria which is reverse CAM positive. Reverse CAM positive, है ना? So 1 bacteria which is reverse CAM positive, that is only bacteria Clostridium perfringens और Clostridium vilchi. एक ही बात है. Only bacteria which is reverse CAM positive. CAM positive और 2 bacteria हम एक पढ़ चुके हैं. कौन सा? Do you remember कौन सा? Streptococcus agalacti. याद आया? Streptococcus hippurate positive, CAM positive. Streptococcus agalacti, that is group B Streptococcus. And along with that, एक और मैं आज ही पढ़ाऊंगी Listeria monocytogenes. Listeria. So these 2 are CAM positive. This is reverse CAM positive. Please write it somewhere. कहीं लिख लो. So all 3 are PYQ, which are CAM positive. So only 2 bacteria are CAM positive. Learn them. Streptococcus agalacti and Listeria. And only 1 bacteria is reverse CAM positive. That is Clostridium perfringens or Clostridium vilchi. Got my point? So please learn that. So it is blood agar. This is Robertson cooked meat media. This is egg yolk agar. And this is reverse CAM. This is reverse CAM. Please learn. So what is the summary? Who will tell me? I'm teaching you Clostridium perfringens. Culture media batao. What you will get on blood agar, you tell me. What you will get on RCM, you tell me. What you tell on egg yolk, you tell me. And one unique reaction, you tell me. So you say, ma'am, on blood agar, we get double hemolysis, also known as target hemolysis. On RCM, we get pink color. That is saccharolytic, not proteolytic. On egg yolk, we get Niegler reaction. And a unique reaction is reverse CAM. No other bacteria in this world is reverse CAM. So for all the bacteria, learn the unique, which is not present in any other bacteria. Please learn. In charon pe PYQ milega. I'm teaching you Clostridium perfringens. है ना? One more important thing is there. One more important thing is that sugar is fermented by all the bacteria. All the bacteria in this world ferment some sugar. है ना? Some ferment glucose, some mannitol, some inulin, alag sugar, है ना? But they all form acid. Out of the sugar, they form acid. Clostridium perfringens is a bacteria. Clostridium perfringens I'm teaching you, no? Clostridium perfringens. Along with the acid, it forms gas also. So from the sugar, 2 things are formed. Acid and gas. It is known as gas-producing bacteria. What it is known as? What is the other name given to Clostridium perfringens? Say gas-producing bacteria. It's a gas-producing bacteria. It produces gas from the sugar. Acid to sab banate hain. All the bacteria make acid, but along with the acid, it makes gas. Do you have any proof? Do you have any proof for this? Prove it. Do an experiment and prove that whatever I'm saying is true. I want to prove that sugar will split into 2 things, acid and gas. Acid and gas by Clostridium perfringens. So Clostridium perfringens split the sugar into 2 things, acid and gas. Let's do an experiment. It is known as milk experiment. Let's take a test tube and fill milk in that. Milk. You know, milk is white in color. Fill it with milk. Fill it with milk. White color milk. You know, litmus paper is available. You know, litmus paper तुमने किए होंगे experiment. So blue litmus convert into red whenever acidic medium is there. Have you read that? Blue litmus convert into red whenever acid is there. Have you read this in your childhood? So the same litmus drops are available. So I'm putting blue litmus drop. Blue litmus drops are available in the market. So I'm putting few litmus drop, the blue litmus, so it becomes blue in color. The milk become blue in color. So simply I have taken the milk. Imagine karo tumne doodh le liya. Take a test tube and fill it milk in that, and put few drops of the litmus. the blue litmus. Now what you are doing, put a drop of the Clostridium perfringens. Clostridium perfringens will go inside. Anna, you know milk contains sugar. The name of the sugar is lactose. Or milk contains sugar which is known as lactose. So lactose will split into two things by this bacteria, the acid and the gas. Acid is formed and gas is formed. Lactose get fermented by Clostridium perfringens into two things, acid and gas. Now you tell me, what does the acid do and what does the gas do? You tell me. What does the acid do? Acid convert blue litmus to red. So in your front of your eyes, you will see the blue color changes to red. As soon as you put one drop, now as soon as you put one drop of the bacteria, the blue litmus changes to red. Number one, this is due to acid. Yeah, acid is doing this conversion. Not only this, whenever we put acid in the milk will coagulate. हिंदी में कहूँ तो दूध फट जाएगा. सुना है दूध फट गया? It will convert into curd. दही के जैसा बन जाएगा. So it will form a clot, a semi-solid clot. This is also due to acid. So acid is causing two things: converting the color blue to red and forming a clot. And the gas which is formed now, first the clot will be formed due to acid, then the clot will be disrupted by the gas. तो भाई पहले क्लॉट बनेगा और फिर फूटेगा. You got my point? रप्चर होके उसमें से गैस बबल्स, you can see the bubbles are coming out, है ना? So ऐसे लगेगा टेस्ट ट्यूब के अंदर तूफान आ गया. It is known as stormy experiment, stormy fermentation. What it is known as? What it is known as? Say stormy fermentation. What it is known as? Just a second. So it is known as stormy fermentation. So this is stormy fermentation, है ना? Stormy clot experiment. You can see the first test tube appreciate the blue color. Appreciate the blue color. You add one drop of Clostridium. As soon as you add, appreciate the color become pink. Color become red. Blue changes to red. Say yes. As soon as this was liquid, you can see it is clot. It is like curd. And you can see the curd is disrupted by the gas. You can see the bubbles. This is stormy fermentation. है ना? Just learn it is a gas forming bacteria. I told you Clostridium perfringens form 12 type of toxin. 4 of them is major, alpha, beta, epsilon and eta, and remaining all are minor. You don't have to learn the names of the minor. Just see there are 12 toxins. Most important is alpha. Alpha toxin से Nigler reaction होती है. Just now I taught you alpha toxin causes the Nigler reaction. Not only this, alpha toxin causes gas gangrene. So alpha toxin causes two things: Nigler reaction as well as gas gangrene. So the disease it is causing is gas gangrene. The disease. Now imagine this is a man. This is a 25-year-old boy, actually a college boy. He was driving motorcycle very rash, and unfortunately he got a road traffic accident. He fell down. He got an injury. He got an injury over the thigh. It is visible. He got an abundant injury over the thigh. He fell in the soil, on the soil. So the wound get contaminated with the soil. He got injury over the thigh. So thigh wound get contaminated with the soil. The soil contains the spores of the Clostridium perfringens. So the spores entered inside. The spores entered inside the muscle, inside the wound. Got my point? The spores will germinate and bacteria will come out. All human have glucose inside our body. मेरे पास भी है glucose. Every human have glucose in our blood. So this person also have the glucose. So Clostridium perfringens will ferment the glucose into two things. So two things will be formed, acid and gas. Got my point? What does the acid do? Can you see blackishness? Acid will cause the gangrene. Imagine acid is fallen down on some skin area. That area will get necrosed, gangrene. Jahan acid girta hai, many face injuries occur because of the acid injury. So acid jahan gira, that area is gone. It's gangrene, it's necrotic. And gas, the gas which is formed, just a second, the gas which is formed, here you can see it is forming the bubbles. Can you see the gas bubbles? It is known as crepitant bubbles. So it is known as gas gangrene. Number one, it is gangrene and it is filled with gas. The gangrenous tissue filled with gas. That's why it is known as gas gangrene. Yes, it will form the blisters. The only treatment is amputation. You have to amputate. Yes, got my point? Got my point? So this person was driving Harsh. I'm telling you, this person is driving and got a road traffic accident. He has a wound. The wound is contaminated with the soil. From the soil, Clostridium perfringens entered in the body. This is the root of the entry. Number one. After getting entry, Clostridium perfringens is degrading the glucose. Glucose is present in the blood ना, तो इसके अंदर भी blood तो है ना, तो इसके blood vessel में जो blood है, Clostridium perfringens is degrading that into acid and gas. Acid क्या कर रहा है? Causing the gangrene. Gas क्या कर रहा है? You can see the bubbles, the crepitant. Got it, everyone? Does anyone have any confusion? So there are 3 bacteria which form gas gangrene, जिस पे question आता है. Please learn. Perfringens तो है ही है, the one which I am teaching you, Perfringens or Wiltshire. Second is Septicum and third is Novi. Learn Punjab से Nagpur, PSN, or make some other mnemonic, है ना? Punjab से Nagpur. Last time some student told me PSN. So you can learn Punjab से Nagpur, PSN, form gas gangrene. Gas gangrene is formed by 3 bacteria. Please, इस पे बहुत question है. Very important PYQ. Which of the following do not form gas gangrene? So you get 3 of these and 1 any other option. So Perfringes, Septicum, Novi. All of them are Clostridium, Clostridium, Clostridium. They form gas gangrene. Please learn it, learn it, learn it. Everyone got it? Harsh, you got it? Others, the one who are saying for repeating? Okay, read the question, tell me the answer. Tell me a non-motile Clostridia. All Clostridia are motile except the Gaddar. Who is the Gaddar? Who is non-motile going against of the family? Who is going against of the family? Not going against with the family. Yes, Perfringes. So all the Clostridia are motile except Perfringes. So go with Perfringes. So it's a very common question. They ask the exception. Examiner love exception. Never forget. Never forget. Examiners always love exception. The next is double zone hemolysis. Double zone hemolysis. Ekmatra bacteria, the only bacteria which show double zone, alpha plus beta, both hemolysis in the same colony. So same colony, first surrounded by alpha, the greenish discoloration, then surrounded by beta, the translucent discoloration. So this is known as target hemolysis. It is seen only in one bacteria, Perfringes. So you all are very correct, very correct. I'm very happy. The correct answer is C. Okay. So we are done with Perfringes. Now I'm coming on the next Titani. There are 4 Clostridium I have to teach you. So let's come on the next Titani. Okay. Let me tell you Titani ke 3 culture media. Hana, what happens on the blood agar? Clostridium Titani. Have you heard about the swarming? Yesterday I taught you swarming. Swarming से क्या याद आता है? Swarming का दूसरा नाम मैंने रखा है PT Usha. है ना? The bacteria which run very fast, the fast runners. है ना? So as soon as you inoculate any fast runner on blood agar, you will see the waves. You will see the waves on the blood agar. Can you see the waves? The waves on the blood agar. है ना? These are the waves on the blood agar. That is known as swarming. The waves, है ना? So I told you yesterday 4 bacteria which show swarming. कौन-कौन से थे? The 2 Proteus, Proteus mirabilis, Proteus vulgaris. The one is Bacillus cereus, and the 4th one is Clostridium tetani. So in Clostridium tetani, the first thing, they are fast runner. They are fast. They are motile, you can see. But you can write here very fast. ये जो लिखा है ना, you can write very fast and you can write इसकी वजह से swarming. इसकी वजह से it is swarming. Motile ये भी है, but they are not fast runner. They are not swarming. Swarming only in tetani. और ये तो non runner है. ये तो run ही नहीं करता. That is non motile. So इसकी खास बात ये it is non motile. इसकी खास बात ये कि it is a fast runner. That is swarming. And ये दोनों are simple runner. Simple walk करते हैं. Say yes, people. I'm trying hard, है ना? So number 1 important point is swarming. It shows swarming, है ना? Clostridium tetani shows swarming. On RCM, it forms black meat. Black. The meat pieces convert into black color. Okay, meat pieces convert into black color. That is, it is proteolytic. Remember, Perfringes was saccharolytic. This one is proteolytic. Got my point? The third is the egg yolk. Egg yolk, there is no zone of opalescence. No zone of opalescence. There is no alpha toxin. Alpha toxin causes zone of opalescence and Niggler positive. This is Niggler negative. So please learn. Hana? So you can see this is swarming. You can see here we have done the inoculation and you can see the waves are formed. This is swarming. So swarming is very unique. Hana? Now listen what I am going to tell you. I am going to tell you something very unique. Have I taught you in your syllabus there are 25 bacteria out of which 10 form toxin? Have I taught you this? Huh? Out of the 10, 8 I already taught you. 8 toxins I already taught you. 4 of them are DPSS. They all inhibit protein synthesis. 4 of them is be very efficient boy. Hana, be very efficient boy. They all increase cyclic AMP. Hana, they inhibit protein synthesis. They increase cyclic AMP. So you don't ask him, ma'am, the total 10 are there now. 4 plus 4, 8. What about the remaining 2? Ab bari hai remaining 2 ki. So now I'm telling you remaining 2 and total 10 toxins are done. One question pakka. Take my words. One question MCQ you definitely get from one of the 10 toxin. It is very important. Ultra super duper important. So the remaining 2 I'm going to tell you. One is here, one is here. Anna, so Clostridium tetani secrete a toxin. The name of the toxin is tetanospasmin. Tetanospasmin. Say tetanospasmin कहते हैं. Clostridium tetani की toxin को क्या कहते हैं? Tetanospasmin. And botulinum की toxin को क्या कहते हैं? Botulinum toxin. Botulinum की toxin को botulinum toxin. So let me tell you the mechanism of action of these two back to back. ठीक है, मैं दोनों का mechanism साथ में बता रही हूँ. Parallel. See. Everyone see. In human body, neurotransmitters are there. Do you have neurotransmitter? We all have 2 types of neurotransmitter. Okay. One is excitatory neurotransmitter and one is inhibitory. है ना? Excitatory neurotransmitter and inhibitory neurotransmitter. There are 2 types of neurotransmitter. Okay. Excitatory neurotransmitter is like acetylcholine. and inhibitory neurotransmitter is GABA and glycine. GABA and glycine. Huh? So excitatory neurotransmitter, what does they do? They cause contraction of a muscle. And inhibitory, what does they do? They cause relaxation of a muscle. See my bicep, my tricep, my deltoid. I can contract, I can relax. I can contract, I can relax. It's my choice. I mean, they are voluntary control. Whenever we want to contract, we secrete acetylcholine at the nerve ending. Whenever we want to relax, we secrete GABA and glycine at the nerve ending. So there is a balance. We can contract, we can relax. All the voluntary muscles say yes. So there is a balance between the two. Normally there is a balance. Both are present, and according to the condition, we can use that. Okay, so let me talk the first bacteria, Clostridium tetani. It secretes a toxin. The name of the toxin is tetanospasmin. Tetanospasmin. What does this tetanospasmin do? Clostridium tetani ka tetanospasmin inhibit the inhibitory neurons. Inhibit the inhibitory neurons. Take my word very clearly. Inhibit the inhibitory neuron. So no GABA, no glycine, no relaxation. So unopposed contraction. No relaxation means unopposed contraction. The body only contract. The full body go in contraction. There is no relaxation. Got it? The name of the disease is tetany. Tetany. What is tetany? Tetany is a disease in which no relaxation and unopposed contraction is there. Hana, what is tetany? It is a spastic paralysis. The complete body go in spasm. Spastic paralysis. Tetany is spastic paralysis. Say people, say yes. Say yes. Coming on the next bacteria. The next bacteria is Clostridium botulinum. The next bacteria is Clostridium botulinum. It secretes a toxin, botulinum toxin. See, this was Clostridium tetani. The name of the toxin is tetanospasmin. This one is Clostridium botulinum. The name of the toxin is botulinum toxin. Botulinum toxin inhibit the excitatory neuron. No excitatory neuron, no acetylcholine. No acetylcholine, no contraction. So unopposed relaxation. The unopposed relaxation. The name of the disease is floppy baby syndrome. Floppy. The complete body go in relaxation. There the complete body go in spasm. Here the complete body go in relaxation. It is known as floppy baby syndrome. Floppy baby syndrome. This one is also paralysis, but flaccid paralysis. It's flaccid. So tetany is a spastic paralysis. Floppy baby syndrome is a flaccid paralysis. I tried my best. I will show you the images, diagram, everything. But have you understood? Have you understood? Say yes, people. So please, can you enumerate the 10 toxin finally? The 10 bacterial toxin. Who will tell me the 10 toxin? Tell me four toxin which inhibit protein synthesis. है ना? Tell me the 4 toxin which increase cyclic AMP. है ना? Tell me 1, 2, 3, 4 here. 1, 2, 3, 4 here. Tell me one toxin which inhibit GABA and glycine. Which inhibit GABA and glycine. One only. And tell me one toxin which inhibit acetylcholine. Only one. So total 10. Total 10. Say it out. So protein synthesis inhibition, Delhi Public Secondary School. Diphtheria, Pseudomonas, Shiga like. है ना? Increase cyclic AMP by stimulating adenyl cyclase enzyme. है ना? And this is inhibiting protein by inhibiting elongation factor 2. पूरी बात बोलो. ठीक है? So here it is be very efficient boy. याद आया? Bordetella, Bacillus anthrax, Vibrio and E. coli. है ना? Inhibit GABA and glycine is tetanospasmin. Clostridium tetani की tetanospasmin. है ना? And acetylcholine वाली Clostridium botulinum की botulinum toxin. Please learn. Take my words, one question pakka. One MCQ out of 300 will be from this page. It's a very big thing I'm telling you. So please don't get confused. Everyone give me a thumbs up. Everyone. Got it? Can we proceed? है ना? So इन दोनों का साथ में हो गया. We have seen the toxins of the two together. Okay. So this is the tetanospasmin. Can you see what does it do? What does it do? Say, ma'am, it inhibits GABA and glycine. It inhibits the inhibitory neuron. No inhibitory neuron, no GABA, no glycine, no relaxation, only contraction. Spastic paralysis, the name of the disease is tetany. See, complete body go in spasm, especially the back muscle. The back muscle. The body cannot relax. है ना? ये जानबूझ के इंसान ऐसा नहीं बैठा हुआ है. देखो, it is not intentional. It is involuntary contraction of the back muscle. The back muscle cannot relax. Involuntary contraction. है ना? So the name of the disease is tetany. The tone is looking like a bow. It's like a bow. देखा है? धनुष है ना? Arrow का धनुष. धनुष आता ना? Right? It's looking like a bow. It is known as opisthotonus position. This position is known as opisthotonus, in which the back muscle go in spasm. It is known as opisthotonus position. ये तो last stage है tetany का. First muscle tell me the first muscle. Here all the muscles are going in contraction. Then this is the complication. Tell me the first muscle. पहली muscle बताओ जो contraction में जाती है पूरी body की tetany में. The first muscle is the jaw muscle. है ना? How do you smile? Can you smile? See, I'm smiling. It's like this. So whenever we smile, the buccinator muscle of the cheeks, it gets contracted. So there is unopposed contraction of the cheek muscle, the jaw muscle, है ना? And this position is known as trismus or lockjaw. Trismus. So this is the first muscle, and this is the complete body going in fast. Officer tonus. So it starts with trismus, that is lockjaw. This is the starting point, and this is the end point. This is the end point. Say yes. So this is the disease tetany. How many of you got it? Read the question. Tell me the answer. One more thing I forgot to tell you. Okay. Here it is written now. The spores are drumstick. Okay, I will tell. ठीक है चलो read the question. I will give explanation here. ठीक है spores with drumstick appearance just now I told you tetany. लेकिन यहाँ पे मुझे एक चीज बतानी है. You can draw. ये चीज notes में नहीं दी. I forgot to tell. If you wish you can draw. There are 4 types of Clostridium. ठीक है. Sometimes the spores are present at the center. No, this is center. Sometimes the spores are present subterminal. This is subterminal. The spores are present subterminal. है ना? Sometimes spores are present terminal. They are bulging. ये सब bulging है spores. लेकिन see, this is central, this is subterminal. अब terminal में it is spherical. Either it is spherical or it is oval. दोनों terminal है ना? So this is central. This is subterminal. This is terminal. This is terminal. They all are bulging. But the location, and here terminal plus spherical, terminal plus spherical, or here terminal plus oval. How many of you got it? Have you got it? See the diagram. You can draw. Please draw. So this shape is known as spindle shape. Okay. This shape is known as club shape. Okay. This shape is known as drumstick. Drumstick का मतलब समझता है? Drumstick. Have you seen a band? In the band there is a drummer. बढ़िया drummer होते हैं, बड़े-बड़े बाल या bald एकदम. Very fashion nowadays. है ना? drummer होते हैं, बढ़िया movies में दिखाते हैं. Very cool. है ना? तो drumstick देखा है? जैसे drum बजाते हैं. Drumstick, है ना? Not piano, not harmonium, drum. है ना? तो जो drumstick होता है, it's like this. So this is known as drumstick appearance. है ना? And this one is known as tennis racket. Have you seen the tennis का racket? तो tennis का racket ऐसा होता है. It's oval. है ना? So this is drumstick. This is tennis racket. Now give the example of one Clostridium in each of them. You have to learn the example. ठीक है जी? है ना? So here learn the example Clostridium bifermentus. Bifermentus. This Clostridium have spindle-shaped spores. Here you write Clostridium perfringens, जो हमारा main hero है, perfringens. That is Clostridium wilchi, gas gangrene वाला. So it formed club-shaped. यहाँ पे याद करो Clostridium tetani, जो अभी हमारा question है. Currently our question is on drumstick, so answer is tetani. And here learn tennis racket is Clostridium tertium. Tertium, है ना? It is also Clostridium. Now where you will get confused, let me tell you. है ना? Both are TT. This is also T. This is also T. है ना? Tertium and tetani, both are TT. One is drumstick, one is tennis. तो कैसे learn करोगे? How you will learn? You can learn. How you will learn? सोचने दो. How you will learn? How you will learn? अच्छा tertium यानी क्या होता है? Third, है ना? First, second, third. Tertiary कहते हैं ना? Primary, secondary, tertiary यानी 3 होता है. Third, है ना? You can learn tennis में third rank आई. So you will not forget. I'm making a mnemonic. कैसे भी याद हो जाए. है ना? Yes. So you can learn like this कि tennis में third rank आई. So tennis is tertium. है ना? And drumstick is tetany. Tetany is drumstick. है ना? You can learn something. Okay. So what I repeat, Gokul, whatever I'm saying, it's written. है ना? So you have to learn the shapes. Spindle shape is seen in Clostridium bifermentus. Club shape is seen in perfringens. Drumstick is seen in tetany. And tennis racket is seen in tertium. You have to learn this. I'm trying to help you how to learn. How to learn. Make some mnemonic and learn. समझ में आई क्या? हाँ टेनिस में थर्ड राउंड याद कर लो, 3 राउंड है ना? टेनिस से टर्शियम याद आना चाहिए. You learn tennis is tertium and drumstick is tetany. You got my point? हाँ ठीक है? Okay. ठीक है. Whatever mnemonic you say यार, but you just learn. Listen now, please. Listen now. Read the question. What I'm saying, drumstick. So drumstick किस में होता है? Drumstick किस में होता है? So answer is tetany. If I change the question from the drumstick to tennis racket, what is your answer now? Would you like to change your answer right now? What is the answer now? Quick, people, quick. If I change the question, what is the answer now? Please, fast. Huh? Yes, in that case, answer tertium. Tennis में 3 round या third rank, whatever. So tennis is tertium, primary, secondary, tertiary. This is how I learned. है ना? So I don't get confused. ठीक है? If I change the question to club shape. What is the answer now? Club shape, answer will become P, Fmge. If I change the question to spindle shape, the answer will become E. So it is not the one question, the 4 questions are hidden here. Please learn the 4 questions. Okay, please learn the 4 questions. Can we go ahead? Haan bolo, say yes. Can I go ahead? Okay. So this is how you have to learn. The sports ka maine bata diya. I forgot to give in the notes. You can make a table. है ना? Just learn. Club shape, spindle shape, उल्टा हो गया. Spindle shape, club shape, है ना? Drumstick shape and tennis का racket shape. Tennis का racket shape. Learn the Clostridium. Clostridium bifermentis, Clostridium perfringens, Clostridium tetani, Clostridium tertium. So यहाँ होता है confusion, tetani और tertium. I told you how to learn. Done. ठीक है? You get questions on that. चलो आगे बढ़ेंगे. So we are coming on the botulinum. Mechanism of action we already know. Botulinum में nothing important. है ना? यहाँ पे culture media में nothing important. Botulinum toxin we have already seen. They inhibit acetylcholine. It inhibits acetylcholine. It inhibits the excitatory neurotransmitter. No excitatory neurotransmitter, no acetylcholine, no contraction, unopposed relaxation. So it is opposite of tetany. The complete body goes in relaxation. Imagine there, the complete body go in spasm. Here the complete body go in relaxation. It is known as floppy baby syndrome. What it is known as? Floppy baby syndrome. It's a flaccid paralysis. Say it's a floppy baby syndrome. So how does it enter the body? I told you tetany enters the body from wound. Here there are 3 ways. Either from the wound, it enters the body, or the canned food. Learn the word canned. Canned food. केन में जैसे फ्राइड राइस से कौन सा हो रहा था बेसिलस सीरस, डेयरी प्रोडक्ट से क्या हो रहा था स्टेफ ऑरियस फूड पॉइजनिंग. यहाँ पे केन फूड से क्लोस्ट्रिडियम बोट्यूलिनम. प्लीज लर्न बोट्यूलिनम. और इट इज प्रेजेंट इन द हनी. कैन यू सी इन सम कल्चर्स बेबी इज गिवन हनी. Honey is considered as very pure or something like that. So babies are given honey. Honey sometimes contains the spores of Clostridium botulinum. So if you are giving honey to any vaccine to any baby, so make sure it is not contaminated. So it is Clostridium botulinum. It is causing floppy baby syndrome. Say yes. No, there is no vaccine available for botulinum. Okay. So can we go ahead? So read the question. Tell me the answer. What is the mechanism of action of botulinum? What is the mechanism of action of botulinum? Can you read the question and tell me the answer? Be fast. Be fast, people. What is the correct answer here? Read the 4 options. Botulinum, I am asking. I told you the 10 toxin. 4411. Yes, you all are right. The correct answer is C. Very good. It inhibits acetylcholine. I am changing the question. Instead of botulinum, if I ask Vibrio, what is the answer now? What is the answer now? What does the Vibrio do? Kahan aata hai Vibrio? You write down. है ना? Delhi private secondary school. Vibrio is not coming. Be very efficient boy. Yes, V for Vibrio. So it increases cyclic AMP. In that case, answer will become A. So you get many questions based on the toxins. Coming on the last one. The last type of Clostridium is difficile. Can I start the last one? The last one is the difficile. So let's start the last one, the difficile. Okay. So difficile में मैं क्या बताने वाली हूँ in the difficile? So listen, we all have colon. Humans have colon. We know the humans have the colon. So this is the colon of the human. See, this is the colon. Okay. Inside the colon, we have good bacteria. We have good bacteria. Okay. We have, these are known as commensal. We know this is present in all of us, me, you, everyone. In all of our colon, we have good bacteria. What is good in them? What good they are doing? क्या अच्छा काम कर रहे हैं वो हमारा? Why they are known as good bacteria? They inhibit the entry of harmful bacteria. They inhibit that. They are like guards. They inhibit the entry of harmful bacteria. ठीक है? Imagine a patient is coming to you. You are a doctor. Imagine a patient is coming to you with some complaint. That doctor, I'm having pus, I'm having abscess, I'm having this, that, ye bhi. So you are giving antibiotic to treat it. Antibiotic. What is the antibiotic? Antibiotic is a drug that kills bacteria. That kill bacteria. Whether good or bad doesn't know. एंटीबायोटिक की आंखें थोड़ी है दिस इज गुड बैक्टीरिया दिस इज बैड मुझे उसको किल करना है इसको नहीं. If you are giving antibiotic, the bad bacteria is also killed, the good is also killed. So if you give a prolonged course of antibiotic to some patient, all the good bacteria in the colon are gone. They are killed by the antibiotic. है ना? So all the good bacteria are gone. So there is no guards now. There is no guards. So Clostridium difficile will get the entry. Clostridium difficile is a harmful bacteria. Normally it cannot enter inside us. It cannot enter inside us because in our intestine, in our colon, the guards are present. And the commensal. But if you give antibiotic to someone, antibiotic will kill the guards. It will kill the good bacteria. Now Clostridium difficile will enter. It will cause the necrosis of the colon. है ना, it will cause the necrosis. So patient presents with diarrhea, number 1, severe diarrhea, number 2 pain, abdominal pain, number 3 यहाँ पे एक membrane बन जाएगी, pseudo membrane, green color pseudo membrane due to the necrosis, है ना, it is known as pseudo membrane colitis. What is the disease known as pseudo membrane colitis या फिर antibiotic associated colitis, antibiotic associated colitis. So you always get a question like that. So there is a patient having some disease. The disease can be anything XYZ dash 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 koi disease. For that you are giving antibiotic. And after the antibiotic, patient have 2 complaints, है ना, patient have diarrhea, patient have abdominal pain. So what is your diagnosis? So typical scenario, antibiotic के बाद diarrhea और pain हो रहा है. So it is pseudomembrane colitis. Same story is written. So if you are giving antibiotic, it will kill the common cell. So Clostridium difficile will enter. It will secrete the toxin and it will cause the necrosis resulting in pseudomembrane formation. See the endoscopy of a normal colon and see the endoscopy of pseudomembrane colitis. Normal colon is normal healthy, है ना? And here you can see the green color pseudomembrane. Green color pseudomembrane. Say yes. So treatment is resection. You have to cut that portion, है ना? और ये तो खुद एंटीबायोटिक से हो रहा है. So what treatment you will give? ये तो खुद एंटीबायोटिक से तो हो रहा है. All antibiotic can do so. So treatment में क्या दोगे? हो ही एंटीबायोटिक से रहा है. You will give metronidazole. Metronidazole, है ना? Metronidazole is an antiparasitic drug. या फिर vancomycin भी दे सकते हैं. ठीक है? So metronidazole या vancomycin is the drug of choice. Okay? Read the question and tell me the answer. We are done with all Clostridia. Read the question and tell me answer. Read it. Okay, chalo. There is a patient having ALL, acute lymphocytic leukemia, having fever, having neutropenia, develop diarrhea after taking amoxicillin. देखो बहुत peculiar है. Patient have diarrhea after amoxicillin. Amoxicillin की therapy के बाद the patient is having diarrhea. The patient is having diarrhea after amoxicillin, है ना? So tell. So it's a very typical scenario. देखते से समझ जाओगे. जैसे menstruating female आया तो तुम देखते से समझ जाओगे toxic shock syndrome. वैसे ही एंटीबायोटिक देने के बाद डायरिया हुआ, सो तुम देखते से समझ जाना इट्स क्लोस्ट्रिडियम डिफिसिल, है ना? नियोनेट देखते से तुम समझ जाओगे द वर्ड नियोनेट सेप्सिस या मेनिंजाइटिस विल इंडिकेट स्ट्रेप्टोकोकस ए गैलेक्टी. सो एवरी क्वेश्चन हैव अ पेक्यूलियर वर्ड. आई वांट यू टू हाईलाइट दैट वर्ड इन योर माइंड कि भाई नियोनेट सेप्सिस आया और ये दिमाग में आए, मेंस्ट्रुएटिंग फीमेल आया और ये दिमाग में आए, एंटीबायोटिक के बाद डायरिया हुआ, एब्डोमिनल पेन आया तो ये दिमाग में आया. So this is how we have to deal with the entire microbiology. These are known as clues. You have to pick up the clues. ठीक है? Clues are very important. Okay जी? Okay जी? So can we move ahead? Clostridia, the 4 Clostridia are done. Huh? So yes, studying sucks. I also agree. Teaching also sucks sometimes. है ना? So studying sucks, but not more than a failure. Imagine the day you are failed and you wish कि I wish उस दिन पढ़ा होता. That is the worst feeling in this world. है ना? So studying sucks, but not more than failure. So studying is good, है ना? I know continuously studying for hours and hours, 10 hours classes every day, it's monotonous. I can understand your feeling, but please remember, it's phasic. It's not forever. It's a phase. It's a phase of your life, and it will pass on. It will pass on. It's hardly for next few months, maybe 2-3 months. That's it. After that, the life is rocking. Believe me. Okay, chalo. So let's go ahead. Let's move to the next bacteria. We are done with gram-positive cocci. We are done with gram-negative cocci. ठीक है? We have started gram-positive bacilli. In the gram-positive bacilli, we have completed bacillus, we have completed clostridium, we have completed diphtheria. Now I'm starting the 4th one, listeria. For listeria, I teach you four lines. That's it. It's a very small bacteria. It's not given in the chart. Listeria के बारे में 4 लाइनें लिख लो, याद कर लो. Listeria. ठीक है? है ना? No capsule. Apply the mnemonic. It is not coming in that mnemonic. Yes, very killer bacteria have pretty nice capsule. It's not capsulated. Spore forming. BSc chemistry में भी नहीं आता. No. Listeria. No capsule, no spore, but it is motile. But it is motile. Have you heard the mnemonic cute baby sleep? हाँ? Cute baby sleep का L क्या है? Cute baby sleep का L क्या है? L is listeria. So it is motile. ठीक है? Which motility? Polar की peritrichus. You can see in the diagram also. It's peritrichus. You can see the tracheas, the flagella is all around. Can you see the flagella is all around? It's peritrichus. ठीक है? So learn one thing. It is having the flagella. It's motile. है ना? No capsule, no sporing, but motility is present. Motility के बारे में दो खास बातें. Number 1, it's tumbling. कल मैंने बताया था tumbling. कोई बताएगा क्या है tumbling का मतलब? What do you mean by tumbling? Jill come tumbling after. Huh? My baby also know what is tumbling. Tumbling is clockwise, anti-clockwise. Can you see? Little bit clock, little bit anti-clock, clock, anti-clock. This is known as tumbling. है ना? So it is showing tumbling motility. Yesterday I make you understand now what is the different type of motility. What is darting? What is tumbling? What is corkscrew? What is swarming? मैंने सबके GIF दिखाए थे तुम्हें, है ना? Yes, clock, anti-clock. So please, only bacteria which show tumbling, only bacteria which show tumbling, it's listeria. So tumbling is a very big clue. Question में tumbling आया, मतलब answer is listeria. That's it. Question में tumbling आया, answer is listeria. That's it. Number one. Number two, motility is differential. What do you mean by differential? It is motile at 25 but not 37. It is differential temperature. At 25, it is motile, but you say 37, non-motile. What is human body temperature? Say, ma'am, 37. So in human body, it's never motile. But outside the human body, it's motile. So I learned that listeria is the bacteria. Listeria, I'm teaching you gram-positive bacilli. I understood it's motile. Motility is present. It's motile. है ना? Motility is present by peritracheous flagella. चलो ये भी लिख लेते हैं. Peritracheous flagella. But there are two things you have to learn here. है ना? Motility के बारे में. Number one, motility is tumbling. It's tumbling. है ना? It's clockwise, anti-clockwise. Clockwise, anti-clockwise, like that. Number one. And it's differential. Number two, it's differential. So it is motile at 25, but not at 37. So learn the two highlighted things on the motility. Learn the two highlighted things on the motility. It's tumbling and it's differential. Say yes, it's tumbling, it's differential. Say yes. So that is the listeria. You can see it's tumbling. Clockwise, anti-clockwise. है ना? The selective media you have to learn here, it's palcum. Palcum. Don't learn the full form, learn palcum. If you want to see the full form, एक बार देख लो, but don't learn the full form. Palcum. P stands for polymyxin. A stands for acroflavin. L stands for lithium chloride. है ना? A कुछ नहीं है actually, है ना? Lithium chloride. C stands for cephalosporin. A is isculin. M is mannitol. Don't learn, है ना? Palcum. You will get palcum only. ठीक है? Palcum is the selective media. And एक चीज बता चुकी हूँ. It is also bacteria which shows CAM positivity. So I told you there are two bacteria which are CAM positive. Which are the two? And there is only one bacteria which has reverse CAM. है ना? I taught you very clearly. So the two bacteria here, one here. I taught you the three bacteria. The two bacteria here which are CAM positive is Streptococcus agalacti और Listeria, जो अभी पढ़ा रही हूँ, Listeria monocytism. ठीक है? And the reverse CAM is Clostridium perfringens. Clostridium wilchi, है ना? So it is CAM positive. Got it? It causes only one disease. Again, neonatal meningitis. Like Streptococcus agalacti only. It is neonatal meningitis. So tell me Listeria के बारे में. Whatever I am asking, you tell me. We are done with Listeria. ठीक है? Listeria की motility के बारे में दो खास बातें. Number one, you have to tell me what are the two unique things. You have to tell me the culture media. है ना? है ना? Selective media, I mean. You have to tell me a unique reaction which is not present anywhere else, biochemical reaction. And you have to tell me the disease, the pathogenesis. The disease it is causing. That's it. Listeria के बारे में 4 point. 1, 2, 3, 4. The chapter is done. The listeria is done. Yes. Motility, say very good, very good. So motility is tumbling. Why not everyone is saying motility is tumbling? Clock, anti-clock, like that. Number two, it's differential. What do you mean by differential? It is motile at 25, but not at 37. है ना? Selective media is palcum. है ना? Unique reaction, it's CAM positive. And disease, it causes only one disease, neonatal meningitis. Hi, I'm done. So Listeria pe you get only these questions. है ना? Now read the question and tell me the answer. Read the question, tell me the answer. There is a patient with a history of consumption of unpasteurized milk, having fever. The blood examination show gram-positive organism with tumbling motility. पूरे क्वेश्चन में काम का एक ही वर्ड है, tumbling. In the entire question, the main word is the tumbling. Only one word is important, tumbling. Based on the tumbling, my answer is Listeria. Whatever is blah, 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 my answer is Listeria. Because it is tumbling. Only one bacteria showed tumbling motility, answer is Listeria. So correct answer is Listeria. Okay. So we are done with Listeria. So we are done with B, C, D, L. And now the next I'm teaching you actinomycetes, the fifth one. So let's start the fifth gram-positive bacilli, actinomycetes. So let's start actinomycetes. Okay. Let's start with actinomycetes. Okay, Ji. So in actinomycetes, actually, listen. Actinomycete is a family inside which two bacteria are there. I will teach you both. So actinomycete mein the two bacteria are actinomyces and nocardia. These are the two bacteria which come under this family. Say yes. एक्टिनोमाइसिटी में दो बैक्टीरिया आते हैं, एक्टिनोमाइसी एंड नोकार्डिया. मैं दोनों पढ़ाऊंगी आपको. I will teach you both. Got it? So one by one we will see both. Please compare the both. है ना? So I'm teaching you actinomycete, which have two bacteria in that, actinomyces and nocardia. Both come under the. What is the unique in that? What is unique? Actino का मतलब पता है? The dictionary meaning actino मतलब होता है rays, है ना? जैसे sun की किरणें आती है ना, rays, है ना? So because these bacteria look like rays, they are like filamentous, both of them. The common property in them, है ना? It is known as filamentous. Both of them are filamentous bacteria. Filamentous bacteria. No other bacteria is filamentous. These are filamentous bacteria, both of them. This is also filamentous, this is also filamentous. Both of them look like fungus, है ना? ऐसा लग रहा है these are the fungal mycelia. So they look like fungus, but they are not fungus. They look like fungus because they are filamentous. They look like so, but they are bacteria. So they look like fungus. They are filamentous. Say yes. Got it? I know. So you can see both of them are filamentous. Both of them look like fungus. Now you tell me. What do you tell me? Both of them are gram positive bacilli. है ना? You tell me obligate aerobe का निमोनिक और obligate anaerobe का निमोनिक. दोनों का निमोनिक बताओ एक एक करके. Obligate anaerobe क्या है? Can't breathe air. तो can't breathe air का A is this. है ना? और obligate aerobe का क्या है? Nasty pest must breathe. तो nasty pest का N is this. I mean to say this one is the anaerobe. Obligate anaerobe. And this one is the obligate aerobe. See, याद नहीं करना है. Don't learn. Mnemonic में दिख रहा है. If you know the mnemonic ना 50% of the microbiology is done. 6 पेज में 60% microbiology है. But मैं 6 पेज देख के बोलती कि खुद ही पढ़ लो तो you people will kill me कि मैम what is this? So I'm explaining you the entire microbiology, but ultimately you have to learn the mnemonics. ठीक है, mnemonics में ही सब कुछ है. Got it? You can see here. A actinomyces है, one is obligate anaerobe, one is obligate aerobe. Say yes. Say yes. So this is the difference between them. No. Both of them are filamentous. This is common. Both of them are like fungus. This is common. But one is anaerobe, one is aerobe. इतना समझ में आई बात? One more difference between them. I told you acid fast bacilli. कल acid fast organism. How many acid fast organism I told you? 10. है ना? Out of the 10, 5 are bacteria, 2 are fungus, 2 are parasite, 1 is miscellaneous. याद आया किसी को? कोई निमोनिक दिया था मैंने एसिड फास्ट ऑर्गेनिजम्स का, ZN ऑर्गेनिजम्स का. याद आया कोई निमोनिक? नो लॉन्गर सेपरेट बुकिंग रिम सिम गार्ड. याद आया? सो नो लॉन्गर सेपरेट बुकिंग रिम सिम डबल कार्ड, जो भी है, N is nocardia. So again nocardia is acid fast, but this one is not coming. This is non acid fast. Nocardia is acid fast. Acid fast में भी तुम्हें फट से याद आना चाहिए मैम, it is an exception. Only 0.5% H2SO4 is required. I told you 3 exceptions. One of them is nocardia. So better to say it's partial acid fast. It's partial acid fast. So till now I have started the chapter actinomycete, and I told you there are 2 bacteria here, actinomyces and nocardia. These are the 2 bacteria. Then I told you both of them are filamentous. Anna, this is also filamentous like fungus. Both of them are like fungus. Anna, so these are the 2 things common in them. Okay. Then I told you there are differences. This one is anaerobe, obligate, and this one is aerobe. We have proven in the mnemonics also. Anna. Then I'm telling you this one is partial acid fast. Partial acid fast. Acid fast to hai, but not complete 20%, only 0.5%. This one is non-acid fast. So these are the differences till now. Do you have any confusion till now? Do you have any confusion? Then I will tell you the diseases. Diseases caused by this, only one disease. Here 3 diseases caused by this. So this chapter is also done. Okay. Okay, got it. So I'm starting actinomycetes, the 2 bacteria, actinomyces and nocardia. So both of them are filamentous. Appreciate the filamentous. This is also filamentous. This is also filamentous. Both of them are filamentous. Anna, they look like fungus. They look like fungus. They are filamentous. They look like fungus. If you take the biopsy, both of them, because of the filamentous appearance, can you see filamentous here also? Can you see filamentous here also? Filamentous. Both of them give sun ray appearance. Sun ray. Have you made sun? Huh? My kid makes sun nowadays. It's like this. Anna, so it's looking like the sun. It is known as sun ray appearance. It is known as sun. So both of them have sun ray appearance. Both of them are filamentous. Both of them are sun ray appearance. Anna, now see the differences. You see the differences. Actinomyces, nocardia. Start. Say the difference. See, ma'am, this is non-acid fast. This is partial acid fast because it is coming in no longer separate booking rim sim card. Rim sim card ka N is nocardia. है ना? This is aerobe. This is, I'm sorry, this is anaerobe, this is aerobe. This is can't breathe air का A, and this is nasty pest must breathe का N. है ना? If you know the mnemonic, कुछ भी याद करने की जरूरत नहीं है ना? Drug of choice दोनों का अलग है, वो तो ठीक है ना? Now let me tell you the culture media of both. Culture media of both. This is the culture media of actinomycetes. This is nocardia. है ना? See, दोनों का culture media कर किस पे रहे हैं? The culture media we are doing on brain heart infusion, BHI. ये भी BHI है. The selective media is BHI. Learn the selective media. It's brain heart infusion, BHI. See the colony. Here the colonies are known as molar. Do you have molar tooth? Molar tooth. The colony is looking like a molar. Molar colony. And here the colonies are looking like cotton wool ball. So say the appearance of the colony. Say, ma'am, here it's molar, spidery molar, and here it's cotton wall. Both of them are BHI. So colony is different. दोनों का colony का appearance बोलो. Please learn the colony appearance. Clinical manifestation one by one. Let me tell you the clinical manifestation of actinomyces. Actinomyces का clinical manifestation, let me tell you. So it causes disease on the face and the neck. Only face and the neck. है ना? Orocervical. Orofacial, only on the face and that cervical area. Only this is. So what happens in the disease? Multiple small sinuses are formed. Can you see? Multiple small sinuses are formed in this area. है ना? If you press the sinus, pus will come. If you press it, pus will come out. They are discharging. Say multiple painless small sinus fistula are formed which are discharging. है ना? If you press, the pus will come. And if you see inside the pus like this, the pus के अंदर छोटे granules हैं. These are known as sulfur granules. Actually, these are the colonies of actinomyces. है ना? So typical discharging sinus containing sulfur granule. Imagine this man. If you press the sinus, pus will come. Pus को ऐसे से करोगे तो you will feel the granularity. It's a very typical disease. No other diseases like this. This is actinomycosis. इसका presentation है. The name of the disease is actinomycosis only. But nocardia causes 3 diseases. Not 1, 3. Number 1, it involves the lung, causes pneumonia. Lobar pneumonia. The complete lobe is gone. Appreciate in the diagram. Severe lobar pneumonia. The complete lobe is consolidated. Okay. Number 2, brain. It causes abscess in the brain. Appreciate the abscess in the brain. Okay. And number 3, same. Hana, multiple sinuses like actinomycosis. If you press them, the pus will come and it is granular. But site, it is not face. It can be anywhere. Hana. So the third is multiple sinuses. So let me confirm. I am done with this chapter. We will solve the question. Let me finalize. I am teaching you actinomycosis, actinomycete, which is having 2 bacteria. Actinomyces nocardia. Can you help me? Say, ma'am, both of them are gram-positive bacilli. है ना? Both of them are gram-positive bacilli. A, B, C, D, L, M का A है दोनों. है ना? Actinomycete है. ठीक है? This one is strict anaerobe, and this one is aerobe. ठीक है? This one is non-acid fast. ठीक है? But this one is partial acid fast. ठीक है? Yes. I forgot to write that, Elizabeth. Yes. So both of them are filamentous. है ना? Both of them are like fungus. Both of them is sun ray appearance. ठीक है? So these are the same things. है ना? And these are the differences you have to see. ठीक है? Culture media बोलो दोनों का. Who will tell me the culture media? It is brain heart infusion having molar colony. Here also brain heart infusion having cotton wool colony. ठीक है? That's it. Now tell me the disease. Here 1, here 3. That's it. Tell me the disease. Finally, last. Here the name of the disease is actinomycosis, in which multiple sinuses formed over the face. They are discharging, having the granule. Here 3 diseases. First, pneumonia. Second, brain abscess. And third is this only, but site is not face. It can be anywhere. Got my point? Respond. Say yes, say no, but give a gesture. Got it, everyone? So let's solve some question. We are done with this chapter also. So last bacteria we will see now, mycobacterium. After that, the gram-positive bacilli is also done. We will move on the last category, gram-negative bacilli. ठीक है? चलो. Madhura foot I will teach you. Yes, Sandesh, don't say Madhura foot here. I will teach you Madhura foot in the fungus and I will correlate it here. Don't worry. ठीक है? चलो. Read the question, take the clues. It's a very important question. Say there is an anaerobe, a strict anaerobe. Number 1, having multiple sinuses, multiple abscess, and multiple sinuses which are discharging, and unke andar sulfur granules are there. What is the correct answer? Everyone is saying A. All those who are saying A as answer. Just a second. All those who are saying A as answer, I agree the answer is A. You all are right. My question to you, why not B? है ना, why not B? B में भी 3 disease हैं. The third presentation is this only ना. Pneumonia, brain abscess. तीसरा क्या था? तीसरा ऐसे ही था. So why not nocardia? I'm asking you. Your answer is right. You are right, actinomycosis. But why not nocardia? Why not? Yes, Elizabeth. Yes, right. No. Sulfur नहीं. The answer is anaerobic. Sulfur granules तो दोनों में होंगे. Sulfur granules will be in both of them. The answer is anaerobe. है ना? So can I say if I change one word in the question from anaerobe to aerobe, what is your answer now? Would you like to change your answer? Huh? I'm changing one word, anaerobe to aerobe. Would you like to change it? In that case, your answer will become nocardia. Because in nocardia, most common is pneumonia, I agree. Second most common is brain abscess, but third most common is this only, no? In that case, your answer will become B. So the clinch in the question is the word aerobe or anaerobe, based on which my answer will be decided. Other things are not important, which are common in both. And so main word is aerobe or anaerobe. I hope you got it. One more question. Take the clues. There is a patient with brain abscess. Okay, brain abscess is a very good clue. है ना? On examination, branching bacteria. Branching matlab filamentous. Branching bacteria which is weak acid fast, partial acid fast, and a gram-positive bacilli is there. Now, what is your answer? What is your answer? Who's saying? Yes, again the answer is A. Huh? Why not B? Why not B? B do not cause brain abscess. है ना? Number one. And it is branching, but acid fast nahi hai. So acid fast and branching is towards the nocardia. If I remove the word brain abscess and acid fast and keep only two words, branching and gram positive, then A and B both are the answers. I hope you all got it. I know. So this is the thing. So let me come on the last bacteria. So we are done with this. We are done with this. And after this, we are done with the third category also. We will move finally on the fourth category. In the fourth category, now only one bacteria is big, Enterobacteriaceae. Rest all are very small. Rest all are very small. Hardly you have to take 2, 3, 2, 3, 2, 3, 2, 3, 2, 3, 2, 3 lines on each of them. You have to learn 2, 3, 2, 3 lines. I mean, very small bacteria are there. So what is the plan now? So I will take all the small bacteria today right now only. Otherwise, if I start with Enterobacteriaceae, these are the big bacteria. Inside it's a big family. So I will cover this only and the time will be over. You will be saying, ma'am, itne sare bacteria bach gaye. Rather, I will cover all and I will start Enterobacteriaceae also if the time allows. Hai By the way, today I will end the session maybe at 5:30, 5:45. Hai because I'm having other commitment. I was not knowing the class will extend. I'm already having other commitment. I cannot avoid it, something unavoidable. So, but tomorrow and day after tomorrow, we are having 2 days more. So we will extend the class tomorrow or day after tomorrow till 7. The next 2 days. It will be till 7 or maybe till 8. I don't have any other commitment tomorrow and day after tomorrow apart from your class. But today I'm having some other commitments, so I'm very sorry for that. So I will end the class today. For today only, it will be 5:30. But tomorrow it will be till 7 to 8, I can't say. And day after tomorrow be depending on the syllabus. But don't worry, in the 4 days, at the end of the 4 days, on 4th of October, your 100% micro is my guarantee. Take my words, it's my guarantee. I will complete. I will not leave even a single word. Whatever I have given you in the notes now, I will complete everything. Okay? So tomorrow and the day after tomorrow, we will extend it. Okay, ji? Chalo. Anyways, है ना? Okay, 10 तक तो नहीं करेंगे, but I will complete the syllabus. ठीक है? जब 10 बज गई तो ठीक है, but I will complete syllabus. Don't worry. But today it's max. It will be 5:30 at max. So it's up to you. Let me teach you mycoplasma, mycobacteria. I mean, after that you tell me. I will give you 2 choices from the gram negative. Gram negative will not be completed today in 1 hour. I require 2 hours here. 2 hours to complete it. है ना? 1 hour entirely for Enterobacteriaceae, nearly 1 or 1.5 hour, and 1 hour for small. So I can complete one. Either I will complete Enterobacteriaceae, not the small bacteria, or I will complete the small bacteria, but not Enterobacteriaceae. Whatever you say, I will complete one of them. One of them will be pending that I will cover tomorrow before starting virology. So first we will cover bacteria only tomorrow. Tomorrow starting also, we will complete bacteria. After completing bacteria, we will start the virology. Okay? Okay, and tomorrow we will complete entire virology. Yes, nocardia can occur on face also, Sasika, but it can occur anywhere on the body. It usually occur on the foot, but I'm not saying no that it don't occur on face. But actinomycosis occurs only on face. Okay, ji. Okay, whatever you say, let me finish first mycobacteria. ठीक है, we will cover all small today, है ना? So बोलने में अच्छा लगेगा कि एक ही बैक्टीरिया बचा है, भले वो सबसे बड़ा हो, पर बोलेंगे अच्छा लगेगा, है ना? Psychologically we feel good कि only one bacteria is left, है ना? But although this one is equivalent to remaining all, let me tell you it's very big. अच्छा, चलो, time wise they are equal. चलो, let's proceed, है ना? Jokes apart, let's proceed. So let's proceed to the next bacteria, mycobacteria. है ना. Mycobacteria, very important bacteria. So let me tell you mycobacterium classification. In the mycobacterial classification, we divide the mycobacteria into three categories. What are the three categories? Mycobacteria tuberculosis complex. All the mycobacteria which cause TB are included in this category. है ना. I will tell you कौन-कौन से bacteria TB कराते हैं. है ना. The second is mycobacterium lepra. They cause lepri. Also known as Hansen's bacilli. And remaining all are fall in the third category. They do not cause TB, they do not cause leprosy. These are known as non-tubercular mycobacteria, NTM. Non-tubercular. So the one which cause TB, the one which cause leprosy, and the third NTM, non-tubercular mycobacterium. है ना? So this is the basic classification. The mycobacterium tuberculosis complex में how many bacteria can cause TB? How many bacteria can cause TB? Most common, 99% is mycobacterium tuberculosis that we will study in detail. The second is mycobacterium bovis. It also causes TB, but not in human. It causes TB in animals like cow, buffalo. And if someone is drinking the milk of such a cow or such a buffalo who is having TB, the person can have intestinal TB. Just suppose I am drinking the milk of a cow or a buffalo or any animal who is having TB, है ना, of mycobacterium bovis. So I can have intestinal TB, है ना. And others में there are many organisms. Don't learn the name. For us, the most important is mycobacterium tuberculosis. So let's start mycobacterium tuberculosis. Let's start mycobacterium tuberculosis. Okay, ji, let's start. Okay, mycobacterium tuberculosis is the most common bacteria which cause TB. TB is a granulomatous disease in which granulomas are formed. है ना, पैथो में भी पढ़ोगी chronic inflammation में. What is the route of the entry? How does the bacteria enters in human body? I'm talking about mycobacterium TB. How does this bacteria enters in human body? Mycobacterium TB कैसे enter होता है? Most common airborne, 99%. It enters via aerosol, है ना. Some person is having TB, the person is coughing, sneezing, है ना, and via aerosol it is entering in another. So first most common route is airborne. Say airborne. Number one. ठीक है. Number two, inoculation, direct skin to skin touch. If someone is having TB and touching another person and direct inoculation, the skin TB can occur. The cutaneous TB can occur. And third is ingestion. I told you some animal is having the TB and you are drinking the milk of that animal. The person can have intestinal TB. So the three routes: airborne, direct contact, and ingestion. Say yes. So these are the three routes by which the TB can enter in human body. ठीक है. Ultimately, it will cause 99% pulmonary TB, 1% extrapulmonary. In the pulmonary TB, the lungs are involved. In the extrapulmonary, any organ in the human body, technically from head to toe, TB can occur in any organ. TB is the only disease जो human के किसी भी organ में हो सकती है. From head to toe, there is no sparing. All the organs can have TB. That is extra, but it's very rare. Mostly it's pulmonary. अच्छा, whatever it is, whether it is this or this, ultimately in that organ the granuloma is formed. Ultimately in that organ, what is formed? Granuloma. What is granuloma? It's type 4 hypersensitivity. Have I taught you hypersensitivity yesterday in this batch? I teach many batches now. I just forget कि किसको क्या पढ़ा है. Have I taught you? I guess yesterday to you only I taught you now. What is hypersensitivity? The 4 types. Another mechanism maybe I have taught you. So type 4 hypersensitivity. Type 4 hypersensitivity. So let me tell you the mechanism. How does the granuloma formed in TB? I'm teaching you the mechanism of tuberculosis. And how does a tubercle or a granuloma is formed? So first of all, the mycobacterium TB enters in human body. The bacteria. Mycobacterium TB नाम का bacteria. This bacteria enters in human body. Usually 99% by inhalational route. So via inhalation route, it is going inside me. For example, it is present in the aerosol. Someone is having TB and coughing in front of me. And it is present in the aerosol. And I am inhaling that. So this mycobacterium TB goes inside me. After going inside me, the first cell it will come in contact is antigen presenting cell, APC. Antigen presenting cell will engulf it and try to degrade it, but it is non-degradable. The unique property, it is non-degradable. It cannot degrade that. So APC will get afraid. It will get scared. कि अब क्या करूँ? ये तो degrade ही नहीं हो रहा. It is not degrading. So let's take it to helper TH1 cell, not helper TH2. Where it will take? It will take to helper TH1 cell. Helper TH1 cell will secrete 3 cytokines from it. It will secrete 3 cytokines from it. What are the 3 cytokines? Interferon gamma, interleukin 1, and TNF alpha. These are the 3. You have to learn. Please learn. 3 cytokines. Now you have to understand the relevance. यहाँ तक तो मैंने कल भी बताया था. Let me continue. These 3 cytokines will take one cell out of the blood. This is the blood vessel. They will take one cell out. Let me start with the first one, interferon gamma. Interferon gamma take the monocyte out. You know, blood have monocytes. Monocytes are one of the WBC. They will take the monocyte out. They will take the monocyte out. Monocyte, after coming out, it is known as macrophage. Monocyte is known as macrophage. Once the monocyte out, it is known as macrophage. Once the monocyte is out, it is known as macrophage. So basically, it is forming macrophage. It will modify macrophage. Macrophage will become large. It will get modified. Modified macrophage is known as epithelioid cell. So it will form epithelioid cell also. What is epithelioid cell? It's a modified macrophage. Multiple epithelioid cell fuses with each other and it will form a big cell known as giant cell. So it is forming 3 cells. And now what are the 3 cells? Say all is done by interferon gamma only. And now number 1 macrophage, then epithelioid cell by modifying and then giant cell by fusing them. Let's come on the next. Who is the next? The next is the interleukin 1. Interleukin 1 will take T lymphocytes from the blood. T lymphocytes out and accumulate it here. T lymphocytes. And finally, the last one, TNF alpha. TNF alpha will take the fibroblast out of the blood and fibroblast will get accumulated. So finally, how many types of cells we have? We have epithelioid cells, है ना? Epithelioid और giant एक ही है, ठीक है? We have T lymphocyte, we have fibroblast. So this will form the center of the granuloma, this will form the middle zone, this will form the outer zone, and ultimately a granuloma is formed. I'm drawing, है ना? So the center is formed by the epithelioid cell. All these are epithelioid cells, ठीक है? Some of them fuse together and they form the giant cell. Ikka dukka. Few of them are giant cell. Okay, this is the central zone. The middle zone is formed by T lymphocyte. These all are T lymphocyte. And the outermost zone is formed by fibroblast. Okay, ji? This is all fibroblast. This is a granuloma. Now take this topic in patho, take this topic in micro, wherever. This is a granuloma. This is a granuloma. See the 3 zones. Now you tell me. Central zone is having epithelioid cell and giant cell. Okay? This is due to which cytokine? You will see, ma'am, interferon gamma. The middle zone में T lymphocyte. It is due to which? Interleukin 1 and 2. And outermost is fibroblast. It is due to TNF alpha. Am I right? So you have to learn in तीनों पे भी PYQ है. Micro में नहीं तो पैथो में मिलेगा. Got it? Yes. You have to learn it. Okay. Now, this is granuloma or tubercle. It is of 2 types. This one is hard because there is no necrosis at the center. This is known as hard tubercle. This one is known as hard tubercle. Sometime at the center, caseous necrosis occur. So if the caseous necrosis is present at the center, it is known as soft tubercle. Right? You have to learn the 3 cytokine and the corresponding cells. वैसे ये topic micro का नहीं है, बस I'm teaching you. It is in patho. Just overlap है तो मैं बता रही हूँ. Interferon gamma is related to epithelioid cell. Interleukin 1 and 2 is related to T lymphocyte, and TNF alpha is related to fibroblast. ये learn करना है. Got it? Harsh, Urva, you got it? You have to learn the 3 cytokines. So this what I mean. Have I written this? I have not written this. So you have to learn the 3 cytokines. Interferon gamma. Nobody is going to ask the question, like diagram on that. The pathogenesis. Interferon gamma. This is the summary you have to learn. Interleukin 1 and 2 and TNF alpha. Okay, tell me. Interferon gamma. Take the monocyte out, convert into macrophage. Modified the macrophage into epithelioid cell and fuse the epithelioid cell to form the giant cell. So ये पूरी कहानी हुई gamma की. Interleukin 1 and 2, they take the T lymphocyte out. And TNF alpha, they take the fibroblast out. So ultimately, a granuloma is formed. Okay, this is how. Got it? Everyone? Urva, Harry, everyone got it? समझ में आई क्या बात? बढ़े आगे? It is of 2 types as I told you. So this is a granuloma. Can you appreciate the granuloma? Appreciate the central blue cell. They are epithelioid. One of them is giant. Appreciate the central red cell. They are T lymphocyte or outermost fibroblast. This is hard granuloma. This is soft. The only one difference is that in the soft, in the center, there is a caseous necrosis. Appreciate the caseous necrosis. So there are 2 types of TB. Primary, pulmonary and extrapulmonary. Pulmonary is 90%. Extrapulmonary is 10%. Pulmonary is again of 2 types. Primary and secondary. Primary TB and secondary TB. Pulmonary TB is of 2 types. Achha. So in pulmonary TB, what is the specimen? What specimen would you like to collect? In pulmonary TB, specimen kya hoga? What is the specimen you will collect in pulmonary TB? What is the specimen we collect in pulmonary TB? Of course, sputum. Pulmonary mein we collect the specimen as sputum. So how many samples of sputum are collected? 2. WHO has given the guideline 2 samples. Imagine a patient is coming to my clinic. Doctor, I'm having cough. I'm having fever. I'm having cough, I'm having weight loss, the three complaint, and the patient is young. I'm having high suspicion the patient is having TB. है ना? So I will, and cough is not resolving with antibiotics. The patient is underweight also. So I'm having suspicion. So I will ask the patient to give two samples. Number one, on spot. Abhi ki abhi. I will give a container and ask the patient to cough and submit it in the container, the sterile container, and submit it to the laboratory. This is known as on spot. On spot sample, number one, है ना, in the OPD only. The second, empty container, you give the patient for taking home, है ना, and ask the patient to give the second sample, first morning sample tomorrow morning, the first sample, है ना. So first is on spot and second is early morning the next day, है ना. Ask the patient to cough and collect it in a sterile container, close the container and submit it to the laboratory earliest. So that is a sample we collect. Now there are 4 types of tests for diagnosis of TB. There are 4 types of tests for diagnosis of TB. Okay. Sandesh, the patient can take the government treatment also and private also. Government treatment is free of cost, the private will be paid. If the government treatment patient is taking, the patient will be put in the DOTS category and given free of cost the complete treatment. And private, the patient can take, but the affordability should matter there. So it is not necessary, you should always refer to the, but TB is a notifiable disease. I guess you want to ask this. TB is a notifiable disease. It's a notifiable disease. You have to notify it to the WHO. So wherever you are working, you just inform to the CMO that I have a case of TB and I am treating it. The patient wants a private treatment, not the government treatment. And you have to inform that. And the CMO will send the weekly or daily report to the WHO. So it's a notifiable disease. The patient can take the private treatment, but it has to be notified. The complete patient details and everything. Got it? It is the case. Okay. So can we go ahead? Okay, ji, hai na? So how many types of lab diagnosis is there? So there are 4. Direct microscopy can be done. We have already seen acid fast staining. Maybe few notes of TB is missing. I have added later on. I have modified it. Maybe old notes are provided to you, hai na, in the booklet. So you can write some extra things, or if you wish, my annotated PDF. Anyhow, I'm going to send you after the class. Later on, after the class, you can write it. Only TB is missing. Other bacteria are not missing. Till now, everything is there. Iske baad bhi sab milega. Only TB mein something is missing because I have modified my notes of TB recently and I forgot to modify in the workbook. Okay, ji? Chaliye. Listen. So the first diagnosis is acid fast staining. Hana, you know what is acid fast staining? Yesterday I taught you. Hana, all bacteria are transparent. Okay, ji. The primary stain is a red stain. Any red stain. It can be carbol fusion like this. So this is also red. This is also red. The mordant is heating, simple heating. After that, decolorizer is H2SO4. Standard is 20% H2SO4. These bacteria are resistant and these bacteria are sensitive. So they maintain the red color. They do not maintain, they lose the red color, they become transparent. And finally, you give blue. So they are still red, they do not accept the blue and they become blue. At the end, we have 2 colors. The red are acid fast and the blue are non-acid fast. The principle is mycolic acid in the cell wall. I have taught you this yesterday. मैंने इसलिए इतना fast बोला because I taught you yesterday. This thing I taught you yesterday. So I guess everyone knows that. So this is acid fast staining. In the acid fast staining, the bacteria look red in color. It is an acid fast organism. The second is fluorescent staining. Fluorescent stains are available. It's Oramin, Rhodamin. Oramin, it's very costly. Here the bacteria appear yellow color. Yellow color bacteria. Can you see the yellow color? It's fluorescent yellow color. Fluorescent yellow color. But it's costly. It's costly. The third is the culture. There are 2 types of culture. Conventional. Culture is the gold standard. There are 2 types of culture. एक तो है conventional, जो हम conventional use करते हैं, दूसरा है automated. One is conventional, one is automated. 2 types of culture. Culture is gold standard. Never forget. है ना? The conventional is Algae media. Conventional. It's the image-based question. The green color algae media on which cream colonies are formed. See, the color of the media is green. Green. It is known as malachite green. कौन सा green है ये? Malachite. Malachite green color है ना? और इस पे color of the colonies is buffy या cream colony. Buffy या cream colony. It's a spotter image. It's the culture media, the selective culture media for the TB. The selective culture media for the TB. Okay, that is conventional. है ना? But growth takes place very slow. It takes 8 weeks. 2 महीने बाद growth होगी. So today I'm having a patient. I will ask the patient to give the culture. 2 महीने बाद report आएगी whether it is positive or not. I cannot wait for 2 months. Based on this, I cannot start the treatment. I mean, बाद में आती रहेगी report for confirmation, but we will start the treatment based on other diagnosis. है ना? And second is automated. Automated means it's a machine like, है ना? ये machine होती है. It's a back tech. इट्स बैकटेक सिस्टम, इसमें फ्लोरोसेंस से क्विंचिंग हो जाएगी एंड आई विल पुट द सैंपल एंड इमीडिएटली विद इन फ्यू मिनट्स आई विल गेट द रिजल्ट. दैट इज ऑटोमेटेड सिस्टम. ठीक है. So we are done with this completely. We are done with gram positive cocci, gram negative cocci, gram positive bacilli. All two, all six. Till now 10 bacteria and the subspecies है ना. स्टेप में हमने 3 पढ़े, स्ट्रेप में हमने 5 पढ़े, निजेरिया में हमने 2 पढ़े, एक्टिनोमाइसिटीज में हमने 2 पढ़े, बेसिलस में हमने 2 पढ़े, क्लॉस्ट्रिडियम में हमने 4 पढ़े. लाइकवाइज हमने बहुत सारे पढ़ लिए, लेकिन हाँ ओवरऑल हमने ये 10 बैक्टीरिया कवर किए हैं. ओके है ना. से यस, दिस इज द वे यू शुड स्टडी बैक्टीरिया. Now I am starting the next last category, gram negative bacilli. So what do you say? Can I start with Enterobacteriaceae or shall I start with Vibrio directly? What do you say? If I start with Vibrio, these are the small, I will cover all. है ना. And if you start with Enterobacteriaceae, only one is covered. Enterobacteriaceae के अंदर ही मुझे बहुत सारे बैक्टीरिया पढ़ाना है. I have to teach you 6 bacteria here. So it will take 1 hour. And I'm having completely 1 hour, 4:30 to 5:30. I will not give any break right now. So can I start with Vibrio? So let me skip. Let me save this much. Otherwise, people will kill me, no? You want annotated, now? Let me save. Just a second. It's saving. Just a second, huh? So let me move on the Vibrio. What is the page number? Meanwhile, you can tell the page number to others. I'm starting with Vibrio. You can skip the 6 bacteria here and come directly jump on the Vibrio. Let's start Vibrio. Okay? Let's start with Vibrio. Okay, start with Vibrio. Are you people there? Can I start? Okay, I cannot see your chat. What is the page number? Page number is 70, someone is saying. Page number is 70. Please open page number 70. Okay, so whatever pages I have left in between, that is complete Enterobacteriaceae. Maybe 4 or 5 pages, whatever is left, I don't know. So I will teach you that tomorrow. Tomorrow I will start in the morning with Enterobacteriaceae only. I will not leave for later on. Tomorrow morning, whatever time we are starting, so we will start with Enterobacteriaceae only. Okay, after that we will continue with virology. Okay, so let's start. Let's start with Vibrio, the next organism. So we have seen this, remaining all is falling in the last category. Let's start with Vibrio. This is the diagram of Vibrio. Vibrio may learn, it's coma shaped. Please learn, very peculiar shape known as coma shaped. कैसे लगाते हैं हम coma? It's coma. It's like coma shaped. Number 1, learn, it's coma shaped. It's gram negative curved rod, it's coma shaped. Number 2, it travel in groups. Have you seen the fishes in a stream? Have you seen how does the fish travel? Fish travel like this in the groups, small groups. Small groups. So in the blood, Vibrio also travel like small groups. It is known as fish in stream appearance. Please learn this. It's a PYQ. Fish in stream appearance because it travels in groups like the fishes in the pond. So it is known as fish in stream appearance. It is coma shaped. Let me tell you the culture media of the Vibrio cholerae. The culture media of the Vibrio cholerae, there are 3 culture media. Let me tell you the 3 culture media of the Vibrio cholerae. Vibrio cholerae is a delicate darling. It's a very delicate darling. So let me tell you the transport media, enrichment media, and selective media. 3 type of media. Since it is a delicate darling, you have to keep it in the transport media. The transport media is Venkataraman Ramakrishnan VR media, number 1. And number 2, Carrie Blair. So learn 2 transport media, Venkataraman Ramakrishnan and Carrie Blair. Learn the 2 transport media. Okay? After that, enrichment media. Enrichment media is alkaline peptone water. It's alkaline peptone water and is Mansoor taurocolate peptone water. It is both of them of peptone water. You know what is enrichment media? It's always liquid. Yaad karo. And selective is always solid. Both of them have stimulatory or inhibitory substances, but one is liquid, one is solid. Okay? So alkaline peptone water and taurocolate peptone water. See the suffix. This is water. This is water. And selective media, you have to learn 3. You have to learn 3 selective media. What are the 3 selective media? BSA, Gatta, and TCBS. If you learn BSA, Gatta, TCBS, it's good. It's good. But see the full form. Don't learn it. Full form learn nahi karna hai. Just ek baar dekh lo. Just have a look. Don't try to mark it up. What is BSA? Bile salt agar. What is Gatta? Gelatin. It's Mansoor gelatin. Taurocolate trypticase telluride agar. Taurocolate trypticase telluride. Triple TA. TCBS is thiosulfate citrate bile salt agar. Let me write. Don't worry, let me write. है ना? Easy करते हैं. चलो, Vibrio cholerae. मुझे तुम बताओ culture media. What is the transport media? What is the enrichment media. ठीक है? And what is the selective media? ठीक है? You have to tell me 2 transport media. You have to tell me 2 enrichment media. You have to tell me 3 selective media. You get many questions from here. ठीक है? Start with transport. You know the meaning. Transport is required for transit because they are delicate darlings. From the collection till laboratory, we have to carry them in that media. Enrichment are liquid media. They are broth. They are water. है ना? And selective are solid media. ठीक है? Selective तो सबके लर्न करने हैं. All 30 बैक्टीरिया. सबके selective की लिस्ट बन रही है तुम्हारी, है ना? You have to read that list. चलो, tell me here. So you are right. It is Venkataraman Ramakrishnan and Carrie Blair. Please learn. नाम याद करने पड़ेंगे. Venkataraman Ramakrishnan and Carrie Blair. VRCB. Learn it. These are the transport media. Enrichment मुझे एक चीज पता है. This is water. This is water. है ना? Now let me fill which water. You will say alkaline peptone water. पहला, it is a water. है ना? और दूसरा मंसूर टोरोकोलेट water. है ना? मंसूर टोरोकोलेट water, alkaline peptone water. The suffix is water. At least you have a clue कि भाई water है कोई ना कोई. Not try to fill कौन सा water है. Say, है ना? इतना difficult नहीं है. Alkaline peptone water, मंसूर टोरोकोलेट water. If you try, it will be easy. ठीक है? Coming on the selective media, learn 3 media. BSA, गट्टा, BSA, गट्टा and TCBS. ठीक है ना? तो BSA क्या है? What is BSA? Bile salt, agar. गट्टा क्या है? Gelatinase, trypticase, telluride, taurocolate, agar. और TCBS, thiosulfate citrate bile salt, agar. Nobody is going to ask the full form. Just BSA, TCA. अगर full form आ भी गया in the option, you will be able to identify. बस इतना करो कि 4 option में you will be able to identify. ठीक है? You will do that. So you get many questions on the media of the Vibrio cholerae. Never dare to forget it. Don't you dare. ठीक है? What is the mechanism of action of its toxin? You will tell me. Mechanism of action of the toxin तुम बताओगे. कहाँ आता है? 10 बैक्टीरिया है ना? Vibrio कहाँ आता है? I'm teaching you Vibrio cholerae. So out of the 10, I told you 4 of them act by inhibiting protein synthesis. उसमें आता है क्या? 4 of them act by increasing cyclic AMP. उसमें आता है क्या? 1 of them inhibit GABA glycine. वहां आता है कि 1 of them inhibit acetylcholine. वहां आता है. कहाँ पे आता है? Where does Vibrio come? I told you the mnemonic ना. Yes, cyclic AMP वाले में. B very efficient boy का V है. तो देखो तुम्हें पता है आधे से ज्यादा चीजें. If you know the mnemonics basics ना, everything is very easy for you. So V for Vibrio. Yes, it increases cyclic AMP. So see, this is the intestine. I'm drawing intestine. The human intestine. ये human की intestine है ना? So these are the cells. Enterocyte. The cells in the intestine I'm drawing. Someone is eating the food which is contaminated with Vibrio. So Vibrio cholerae is going here. This is Vibrio cholerae. It is there in the lumen of the intestine. Vibrio cholerae will secrete a toxin known as cholera toxin. कौन सा toxin? Cholera. Vibrio cholerae will not go inside the cell. The cholera toxin will go inside the cell. Cholera toxin. See, bacteria do not go. The toxin secreted by the bacteria go. Say yes. The toxin secreted by the bacteria going inside the cell. After going inside the cell, it is stimulating adenyl cyclase. It is stimulating adenyl cyclase. Adenyl cyclase convert ATP to cyclic AMP. More and more cyclic AMP will form. Say yes. More and more cyclic AMP will form. So what does the cyclic AMP will do? On the surface, sodium channels are present. These are known as sodium channels. Cyclic AMP inhibit the sodium channel. आगे की कहानी सुनो. यहाँ तक तो सबको पता है. Everyone knows still cyclic AMP. Cyclic AMP inhibit sodium channel. So sodium will not be reabsorbed. Normally sodium is reabsorbed in the blood. But here the sodium channels are blocked. So sodium will remain in the lumen only. Sodium will not reabsorb. Because it is blocked. The sodium channels are blocked by cyclic AMP. Sodium के पीछे पीछे जाता है water. Wherever sodium go, water goes there only. It's a rule. So no sodium absorption, no water absorption. Both of them will remain in the lumen only and they cause diarrhea. They cause diarrhea because they are not reabsorbed. So they will cause diarrhea. Say yes, people. How many of you got it? How many of you got it? Respond fast. Respond fast. Got it? So the same story is written in front of you. Okay? Can you see? This is cholera toxin, not bacteria. The toxin. The toxin is entering in the heart. I forgot to tell you the receptor. It is GM1 ganglioside. I forgot to tell you these are the intestinal cells I told you. No? These are the intestinal cells. This is Vibrio cholerae. Vibrio cholerae secrete a toxin. The toxin is cholera toxin. So this cholera toxin enters in the enterocyte with the help of a receptor. The name of the receptor is GM1 ganglioside. Is PYQ hai. GM1 ganglioside. Please learn it. So it is GM1 ganglio. Via this GM1 ganglioside, the cholera toxin enters inside. After entering inside, it stimulates adenyl cyclase. है ना, it stimulates adenyl cyclase. Adenyl cyclase convert ATP to cyclic AMP. Cyclic AMP increases. It inhibits sodium channel. So sodium will not reabsorb. Water will not reabsorb. It will cause diarrhea. Say yes. Sodium and water will remain in the lumen only, and in the lumen they will cause diarrhea. Got it? This is the mechanism of action. Okay. So patient have only one complaint: diarrhea, diarrhea, diarrhea. Patient have only one complaint. थोड़ा बहुत vomiting भी होगा, but main is diarrhea. It is known as typical watery diarrhea. Highlight the word watery. Patient will say कि doctor साहब, I am having severe watery diarrhea. There is no solid content in the stool. It's only pure water. Watery diarrhea. Watery diarrhea. Yes. So what is the color of this? Is it yellow? Say no. Is it cream? No. Is it buffy? No. What is the exact color? Transparent, translucent? No, no. Have you ever tried washing the rice before cooking? Have you cooked rice ever? Huh? Before cooking, we wash the rice now. So have you seen the color of the water in which we wash the rice? It's looking like that color. I know the synonymy is not very good, but it's given. So you have to learn. It is known as rice water stool. Because of the color, it is known as rice water. It is something between cream, buffy, like that. So you will say it's rice water stool. It's a very big clue you get in the question. Rice water stool, watery diarrhea, Vibrio cholerae. Fever is absent. You know the mechanism of action. Fever is absent. So we are done with Vibrio. Okay, Vibrio में क्या क्या सीखा? चलो, let's summarize. छोटे छोटे बैक्टीरिया हैं Vibrio. In the Vibrio, in the morphology, you have to tell me something. Okay, morphology में say capsule है कि नहीं? Say spore है कि नहीं? No capsule, no spore. But motility is something unique. You have to tell me, है ना? उसके बाद you have to tell me the shape. यहीं पे shape भी बता देना. इसका shape, है ना? और उसका appearance in blood. So this is all morpho. ठीक है? Morpho के बाद आ जाओ culture media पे. Culture media में you have to tell me transport media. You have to tell me enrichment media. And you have to tell me selective media. है ना? You have to tell me 2 transport, 2 enrichment and 3 selective. Total 7 media. ठीक है? कल्चर मीडिया के बाद मैकेनिज्म ऑफ एक्शन ऑफ टॉक्सिन. ठीक है? एंड उसके बाद डिजीज. दैट्स इट. इतना ही तो पढ़ाया है मैंने. ठीक है? चलो शुरू करो. नो कैप्सूल, नो स्पोर. मोटिलिटी इज प्रेजेंट. मोटिलिटी इज प्रेजेंट. विच मोटिलिटी? वेरी पोलर सोल्यूशन HCl. याद आया क्या निमोनिक? वेरी पोलर सोल्यूशन HCl. से मैम मोटिलिटी जब भी बोलो तो बोला करो पोलर की पेरीट्रैकस. Now at your stage I am not expecting it is motile ma'am. Along with the motility, you should tell whether it is polar or peritracheous. Now you are grown up, na? You are not small kids now. ठीक है ना? Brushing हो गई ये micro की. So you should say always, is it polar or peritracheous? So it's polar. It is coming in which? It is not coming in cute baby sleep. It is coming in very polar solution HCl. So it's polar. ठीक है? Regarding the shape, coma shaped. है ना? Regarding the appearance, fission stream appearance. Fission stream in सब पे question है. In सब पे question है. ठीक है? Transport media we have seen. Transport media Venkataraman Ramakrishnan and Carrie Blair. Enrichment both are water. है ना? One is alkaline peptone water. One is Mansoor taurocolate water. Selective media भी मैंने बता दिया. BSA है ना? गट्टा है ना? BSC हुआ, गट्टा हुआ और तीसरा क्या था? What was the third one? I'm skipping. हाँ, TCBS. ठीक है जी? So please learn TCBS है ना? Mechanism of action of the toxin, just now I taught you, it increases cyclic AMP in the intestinal cell. Cyclic AMP increase in the intestinal cell, so it will inhibit the sodium channel. No sodium absorption, no water absorption. It will lead to watery diarrhea. The name of the disease is watery diarrhea. It is known as rice water stool, watery diarrhea. Yes, the receptor is GM1 ganglioside. Okay. So can you proceed? है ना? ये सब हुआ विब्रियो के बारे में. The motility I forgot to tell. Darting motility. है ना? The motility is darting. What is darting? Who will tell me the meaning of the darting? I told you yesterday. Shooting star. It's shooting star. It's as fast as shooting star. टूटते हुए तारे जैसा fast होता है. So this is also very unique. So read the question. Tell me the answer. We are done with vibrio. छोटे छोटे से बैक्टीरिया हैं, फटाफट होंगे. Fast. 5 minutes on each bacteria. Okay. Why can't we see rice water stool? Here the complete canal, the complete enterocytes are blocked. Maybe in other bacteria, the complete enterocytes are not blocked. Maybe some solid content is there, but here there is no solid content, only liquid content. Okay. Rice water stool seen only in Vibrio. It's a very hallmark. Yes. So there is a lady coming to the hospital with diarrhea. So first clue given to me is diarrhea. There is a lady having diarrhea. She is attending Kumbh Mela. I mean overcrowded place. Kumbh Mela का relevance है कि she is coming from the overcrowded place. Maybe some contaminated thing she has eaten. है ना? Now the organism is shown in the image. So first identify the organism. ना तुम सबने आंसर बोल दिया darting. Just now I taught you. That's why. If you all 19 subjects are taught to you, then this question is given to you. No, the same question becomes difficult. I always say two things: see the shape, see the color. Take my words very seriously. Take the shape, take the color. See the shape, see the color. What is the color? Is it red or violet? Of course, it's red. You can see these all are red. So it's a gram-negative organism. मुझे पता चला it's gram-negative, है ना? And what is the shape? It is bacilli. Of course, these are elongated. These are not cocci. So it's a gram-negative bacilli, है ना? Gram-negative bacilli में typically yes, these are coma-shaped. Coma-shaped is vibrio, है ना? एक clue, it's coma-shaped. These all are coma. You can appreciate the coma. Appreciate the coma. Most of them are coma. Number one. Number two, one more thing. It is having the polar flagella, and most of them I can see the polar flagella. One polar flagella. Polar flagella किस किस में होता है? Very protective solution HCl. इसमें से डायरिया कौन कौन कराता है? So इसमें से डायरिया विब्रियो कराता है, and it is having darting motility. So this is the approach. This is the approach. I can see a polar flagella. Polar flagella 6 organisms हैं. Very polar solution HCl. HCl. So vibrio is having darting motility. Say yes. Got it? Can we proceed? है ना? So we are coming after vibrio. The next organism, Legionella. Legionella में only 3 things are important. Learn 1, 2, 3. That's it. Legionella is very easy organism. छोटा सा organism है. Let's start the next organism, Legionella. Legionella का selective media is important. Selective media is BCYE. है ना? You are completing your table. I am assuming जो मैंने general में table दिया है. You have to follow that table. So here the selective media is BCYE. What is the full form of BCYE? It's buffered. B is buffered, C is charcoal, है ना? It is yeast extract. So buffered charcoal yeast extract. इसमें 3 चीजें हैं. एक तो इसमें buffered charcoal है और एक yeast extract. 2 चीजें हैं. So buffered charcoal yeast extract है ना? It is one of the bacteria which is capnophilic. It is capnophilic. It do not require oxygen. It require carbon dioxide. It is known as capnophilic. Capnophilic. Only 2 bacteria are capnophilic. एक तो ये Legionella and one is Brucella. Brucella is also capnophilic. So these are the 2 bacteria which require 5 to 10% of carbon dioxide. Let me enumerate. चलो revise करेंगे. We will revise. Tell me strict anaerobe. है ना? I mean obligate anaerobe. Tell me the obligate aerobe. Tell me the obligate aerobe. I will tell only mnemonic, not the full form. है ना? Don't worry. Tell me microaerophilic. Microaerophilic. What is microaerophilic? The one which require 5 to 10% oxygen. Okay, ji? Microaerophilic. And tell me capnophilic. Capnophilic. Anna, the one which require 5 to 10% carbon dioxide. Now see, this is oxygen, this is carbon dioxide. Tell me the bacteria. So here you will see can't breathe air. Everyone knows. Nasty pest must breathe. Everyone knows. Hell. Camp. Boring. Hell, the camp is boring. ये तीनों मैं अभी पढ़ाऊंगी 5 minutes. छोटे छोटे बैक्टीरिया हैं. We will complete. So I will not say it again. मैंने एक ही बार में बता दिया. Helicobacter, Campylobacter. After Legionella, my next 2 bacteria are these only. है ना? So Helico and Borrelia will be the last bacteria. ठीक है? चलो. और Capnophilic में this one Legionella. Legionella, which I am teaching you right now, and Brucella. Brucella. Make some mnemonic, ना? BLLD. कुछ बना लो. BL, है ना? So make some mnemonic. Can't breathe air. Nasty pest must breathe. Hell, the camp is boring, and LD. So these all are based on oxygen and carbon dioxide. Okay, G. You get many questions from this. So see, the peculiarity is that I'm teaching you Legionella. Vibrio is done. I'm teaching you Legionella. Legionella mein we have learned 2 things. Number 1, the selective media is BCYE. What is BCYE? Buffered charcoal yeast extract. And number 2, it is capnophilic. It requires 5% carbon dioxide, not oxygen. ये 2 चीजें पढ़ी. Number 3, the diseases it causes. It causes 3 diseases. Learn all 3 diseases. Number 1, it causes Pontine fever. है ना? Pontine fever is हल्का फुल्का flu. है ना? सर्दी खांसी जुकाम. No treatment required. No pneumonia, no treatment required. Little bit of fever, headache and malaise. That's it. Or running nose. That's it. Or treated automatically. No treatment required. Pontine fever. But you get a PYQ. Pontine fever is caused by. So answer is Legionella. Answer is Legionella. The second is Legionnaire disease. Here, severe pneumonia will occur. Treatment is required. Patient has cough, dyspnea, fever, diarrhea. But you don't get question on that because it is obvious कि Legionnaire disease is caused by Legionella. इतना easy question नहीं आएगा exam में. You will get question Pontine fever is caused by. तो तुम सोचोगे क्या है? but legionella disease is caused by legionella, obviously. So this question will not come. Extrapulmonary, ये भी lung. Extrapulmonary में most common organ is heart. So these are the three diseases. So in legionella, we are done. Legionella, we have learned three points. 3 points you have to learn. Number one, learn the selective media. You will tell me selective media. ठीक है. Number two, oxygen और carbon dioxide का क्या unique thing है? Tell me something unique there and tell me the name of the diseases. The three diseases it causes. 1, 2, 3. That's it. Complete. है ना? चलो. छोटे-छोटे से बैक्टीरिया हैं. Selective media, please learn BCY. है ना? BCY. Buffered charcoal yeast extract. It is a capnophilic. It requires 5% carbon dioxide, not oxygen. It is capnophilic. That is unique in that. Unique क्या है वो learn करना है. And the three diseases it causes. What are the three diseases? Pontike. Pontike पे very important PYQ. Pontike fever, legionnaire disease, and the third one, extrapulmonary organ is heart. Extrapulmonary organ is heart. Please learn. So these are the three diseases. इतना सा ही बैक्टीरिया है legionella. छोटे-छोटे नोट्स बनाते जाओ. और I have given it, just compact it and learn. Legionella. ठीक है? So read the question, tell me the answer. चलो. So there is an elderly man presented with fever, presented with cough. The patient presented with fever and cough. Okay, pulmonary symptoms are there. Sputum show a gram-negative organism. Something gram-negative. है ना? They all are gram-negative, by the way. They all four are gram-negative. I cannot rule out anything. है ना? But it can grow on BCY agar. So the biggest clue, according to me, in the question is only one. The biggest clue is BCY. है ना? BCY. BCY is selective only for one bacteria, Legionnaire. So you see, you should know the selective media of all 25 bacteria in your syllabus. All means all. Okay, so that is the Legionella. Can we proceed? Can we proceed ahead? Once you believe, you are already halfway there. है ना? Believe तो करो. आधा रास्ता तो ऐसे ही साफ हो गया. है ना? Just on the belief. After that, your hard work and your knowledge and everything else matters. The first thing, you believe in yourself that you are going to crack your exam this time. First, have a belief in yourself that you are going to crack this time. Not other time, not second shot, this shot. This shot is the final shot. And you are going to crack that. You are among the 10% or 20% results. You are among that. You are in. You are already in. Okay, believe. Okay, chalo. Anyways, so motivation ka itna time nahi hai. I'm having many motivational videos also, small clips. But I don't get time to show you. है ना? Anyways, chalo. So we are done with Vibrio. We are done with Legionella. Enterobacter C, we have kept it on hold for tomorrow. ठीक है? So coming on the next one, Helicobacter, and followed by Campylobacter. I call them brothers. Another twin brothers. साथ में ही आते हैं हर जगह. Helicobacter, Campylobacter. So let's start first with Helicobacter, and then we will move on Campylobacter. तो ये दोनों भी हो जाएंगे. So let's start with Helicobacter. है ना? ये सब छोटे से बैक्टीरिया हैं, फटाफट हो जाएंगे. ठीक है? चलो, let's start with Helicobacter. So Helicobacter pylori. पूरा नाम क्या है? Helicobacter pylori. ठीक है? No capsule, no spore, but motile. अच्छा. Very protective solution HCl. देखो मैं दो निमोनिक बताती हूँ. Very protective solution HCl. ये निमोनिक किसका है? Polar flagella. Polar flagella. ठीक है कि नहीं है? So Helicobacter, Campylobacter both are coming here. I told you ना, both always come together. एक और निमोनिक बताती हूँ. Hell camp is boring. Hell camp is boring. ठीक है? ये निमोनिक किसका है? ये निमोनिक किसका है? This mnemonic is of microaerophilic bacteria. Microaerophilic. So here also see both are coming. So they both come together. I mean to say both of them are motile. Both of them have polar flagella. Both of them require 5 to 10% 5 to 10% oxygen, not carbon dioxide. Carbon dioxide Legionella और Brucella में लगेगा. यहाँ पे oxygen लगेगा. है ना? So please learn that. Okay? Yes, micro. So both are microaerophilic. So you can see both of them are motile. It's motile. दोनों का shape same है. इसे कहते हैं flying bird. Flying bird. Birds are flying like this, ना? Flying bird shape. इसे कहते हैं seagull. What is the shape? Seagull. Seagull is a bird. Seagull shape. है ना? So coma shape कौन था? और seagull shape कौन है? You tell me. Coma shape कौन था? Vibrio. है ना? और seagull कौन है? Helicobacter, Campylobacter both. Both of them are seagull. So appreciate. Write down the word seagull shaped. Seagull. They are gram-negative seagull. Highlight the word seagull. है ना? Highlight. You can appreciate ये seagulls. ये ऐसे birds. Seagull. So seagull shape is there, motile is there, micro. हाँ, selective media दोनों का skew है. Both of them. The selective media is skew. Everything is common, only disease is different. ठीक है, skew. Skew media, very unique media. Skew media is selective for both of them. Okay? Oh, got it? है ना? and it is microaerophilic. I already told you, no, microaerophilic. It requires 5% oxygen. ठीक है? Diseases. So I'm teaching you the diseases of Helicobacter, and after that I will teach you the diseases of Campylobacter. ठीक है? Helicobacter. The Helicobacter causes 7 disease. 7 disease करता है. सातों के सातों एक ही organ में stomach. All the diseases are in stomach. It involves only one organ, that is stomach. It involves only one organ, that is stomach. All 7 diseases. So 5 of them are benign. 5 of them are benign and 2 of them are malignant. I mean to say, I will teach you 25, 30, whatever bacteria. Only one bacteria is oncogenic. What do you mean by oncogenic bacteria? Which cause cancer, and that is H. pylori. H. pylori is the only bacteria which is oncogenic among all the bacteria in the world. It causes cancer. It is a cancer. I told you many bacteria till now. Na? I taught you staph, strep, Neisseria, Bacillus. कितने सारे पढ़ा दिए मैंने 10, 15, 20 तो पढ़ा दिए. उसमें कहीं भी कैंसर बताया मैंने? I have not taught you cancer anywhere. First time I am teaching you a bacteria causing the cancer. है ना? So it causes 2 cancer. Both of them are in stomach. One is adenocarcinoma of the stomach. One is adenocarcinoma of the stomach, and second is lymphoma of the stomach, known as maltoma. Lymphoma of the stomach is maltoma. Say, so adenocarcinoma and lymphoma, these are the 2 cancers it causes. Okay, so there are oncogenic organisms. Oncogenic organisms, all the organisms which cause cancer. है ना? So total 11 or 12 organisms are there. ठीक है? 1 bacteria is there, which I have already taught you. There are 7 viruses and there are 4 parasites. है ना? 1 bacteria I already taught you, it's H. pylori. H. pylori is the only bacteria which cause cancer. है ना? 4 parasites I will teach you in parasitology tomorrow. It's Fasciola hepatica, Clonorchis sinensis, Cystosoma, and Opisthochus. These are the 4 parasites. Viruses EBV hai, HPV hai na? There are many. HTLV hai. There are many viruses. So we will study tomorrow. Viruses जब पढ़ाऊंगी ये पढ़ाऊंगी, parasite जब पढ़ाऊंगी tomorrow and a day after tomorrow. Don't worry. But bacteria में से it's only one. H. pylori. It causes 2 cancer. कौन से 2 cancer? Both of them stomach. एक adenocarcinoma of the stomach, एक maltoma of the stomach. Say yes. समझ में आई क्या? Say the 5 benign diseases. It gastritis. Say ma'am, acute gastritis, chronic gastritis, autoimmune gastritis. तो 3 तो gastritis हो गए. उसके अलावा peptic ulcer. Gastritis leads to peptic ulcer. So उसके अलावा हो गया peptic ulcer. And last is pernicious anemia. Pernicious anemia क्यों होता है? You know why pernicious anemia occur? So see, this is the stomach. In the stomach we have parietal cell. You know we have parietal. Chief cell भी होते हैं, parietal भी होते हैं. Parietal cells secrete intrinsic factor. Intrinsic factor. This is intrinsic factor. So whatever we eat now, vitamin B12 in diet. So vitamin B12 go inside. This vitamin B12 form a complex with intrinsic factor. And then only the assembly is absorbed. Alone vitamin B12 is not absorbed. है ना? Imagine someone is having H. pylori gastritis. In H. pylori gastritis, the parietal cells are destroyed. So if parietal cells are destroyed, intrinsic factor will not secrete. So person is eating B12, but B12 will not absorb because intrinsic factor is absent. Such anemia is known as pernicious anemia. So vitamin B12 deficiency will occur. It will lead to pernicious anemia. So if you got the mechanism, it's good. If you didn't got it, वो पैथो के hematology में फिर से पढ़ोगे. ठीक है? चलो, कोई बात नहीं. Yes, unable to absorb vitamin B12. Yes. So tell me the diseases caused by H. pylori. Who will tell me the diseases caused by H. pylori? Tell me 5 benign and 2 malignant. Total 7 diseases. Say, tell me 2 malignant first. 2 malignant. Say, ma'am, adenocarcinoma of stomach and say maltoma of stomach. Tell me the 5 benign. Say acute gastritis, say chronic gastritis, say autoimmune gastritis, say peptic ulcer, and say pernicious anemia. If you can learn, it's good. So these are the diseases caused by that. है ना? What is the test? What is the sample specimen you will take? Specimen क्या लोगे? I'm asking for H. pylori. H. pylori में specimen क्या लोगे? I'm asking. जल्दी बोलो, फटाफट. Huh? What is the specimen you will take? Patient is coming to me. So just suppose there is a patient in my clinic. Patient is complaining of acidity. Patient is having pain in the abdomen, acidity, dyspepsia. Yes, it's biopsy. There is no other specimen. You have to go inside and cut a chunk from here and come out. Endoscopic biopsy. So you have to do the biopsy. है ना? Biopsy करके क्या करें? Biopsy करके you can do the gram staining. You will see seagull morphology. है ना? You can do the culture. Which culture? Skews. I don't know the spelling. It's skews. है ना? Skews media. gram culture कर सकते हो, skews, and there is a special stain. Learn the special stain. It's Warden's-Starry. Warden's-Starry. Warden's-Starry give brown color to the H. pylori. H. pylori will get highlighted. जहां जहां H. pylori वो highlight हो जाएगा biopsy पे. ये तीनों चीजें हम कर रहे हैं biopsy पे. ये तीनों की तीनों चीजें काय पे कर रहे हैं हम? All the three things we are doing on biopsy. Say yes. All the three things we are doing on. All the three things we are doing on biopsy. है ना? I'm sorry. So all the three things we are doing on biopsy. Okay, ji, है ना? Now, biopsy patient don't like biopsy because it is invasive. Na, you will ask the patient, we have to admit you. We will give you anesthesia. We will put an endoscope inside you. We will cut a portion of your stomach and come out. Patient will run away. It's very costly also. Patient will say, no, मुझे नहीं कराना. थोड़ा बहुत पेट में दर्द है, a little bit of abdominal pain, and you are asking to cut inside my stomach. No, I will not do that. Patient don't like invasive test unless until it's not cancer. Patient don't like that. So tell me some non-invasive test. किसी non-invasive तरीके से हम कोई sample ले सकते हैं, कुछ कर सकते हैं, जिसमें biopsy ना लेना पड़े. Is there any non-invasive way? Is there any non-invasive way? Yes. There is urea breath test. Please understand, it's difficult and very important. Urea breath test. Imagine what I will do. This is my patient. No, this is my patient. I am suspecting my patient is having H. pylori infection. I do not want to do the biopsy. Okay, I will ask the patient to drink urea. Urea is available commercially. Urea. This urea I will ask in a fixed dose. Whatever is the fixed dose, I will ask that ml of the urea to drink it. Urea is available commercially. I am doing this test. This is not normal urea. You know the chemical formula of urea? Chemistry में पढ़ा होगा. I don't remember, but it contains carbon. ये तो sure है. This carbon is radioactive. It's C13. It's not C14. Normal carbon is C14. This one is C13. So I'm asking the patient to drink radioactive urea. This is urea breath test. So patient will drink it. Patient, the urea will go inside like this. Urea is there in the stomach. These are the cells of the stomach. Can you see? These are the cells of the stomach. Now there are two possibilities, whether the person is having H. pylori infection or do not have H. pylori infection. That I want to know. So just suppose the person is having the H. pylori infection. Here is the H. pylori over the cells of the stomach. H. pylori is lying down. H. pylori is over the cells. The mucus layer वहाँ पे पड़ा हुआ है. So this is H. pylori. If H. pylori is present, it secretes enzyme urease. What does it secrete? H. pylori secretes enzyme urease. Urease will split the urea. So what does this urease will do? This urease will split the urea. It will degrade the urea. Urease will degrade the urea into two components. Urease will degrade the urea into two components. Number one, ammonia. Number two, carbon dioxide. Urease. Urease is coming from the bacteria. So both ammonia and carbon dioxide will go in the blood. These are the end products. So they both will go in the blood. Ammonia and carbon. So ammonia will go in the blood and excreted from the kidney in the urine. Uska tension nahi hai. Ammonia will be excreted in the urine from the kidney. What about the carbon dioxide? This carbon dioxide contains the carbon. कौन सा carbon? ये carbon dioxide कहाँ से आ रहा है? The carbon dioxide is coming from the urea. But in the urea, the radioactive carbon is there. So this carbon dioxide have radioactive carbon. This is radioactive carbon dioxide. You got my point? Carbon dioxide is not excreted in the urine. The ammonia is excreted in the urine, but carbon dioxide is excreted from the lungs. I'm inhaling and I'm exhaling carbon dioxide. So carbon dioxide is excreted from the lungs. So it will go to the lungs. So this carbon dioxide will go to the lungs of the patient and patient will exhale it out. This carbon dioxide will exhale in the breath. So we have an instrument known as breath analyzer. एक छोटा सा instrument होता है breath analyzer. So we will ask the patient to exhale in that like this. We will put it in the mouth and ask the patient to exhale. So if the patient will exhale and if it contains radioactive carbon dioxide, it will show the flickering. It will show the buzzer. So we will come to know that it is having the radioactivity. Say yes. So we will come to know why radioactivity is there, because it is secreted by the urease. और urease कहाँ से आया? H. pylori से. Imagine अगर H. pylori नहीं होता. If the H. pylori is not there, so urease is not there. Urease is not there, urea will not split. Urea will remain as urea only, and urea is excreted in the feces. It will be excreted. So in the breath, you will not get carbon dioxide, the radioactive carbon dioxide. This is urea breath test. And now why it is known as urea breath? Patient is doing two things, drinking urea and breathing. Urea, breathe. Ask the patient to drink urea and after few minutes, ask the patient to exhale like that. Urea breath test. It's a non-invasive test. We don't have to take the biopsy. Say yes. So this is urea breath test. How many of you got it? है ना. So this is H. pylori. ठीक है. Read the question, tell me the answer. Endoscopic biopsy is positive for urease test. And the organism grows on skew media. Identify the organism. How many clues are given to you? The organism is urease positive, urease test, and skew media. What is the correct answer? Tanu, Elizabeth, Saju, everyone is saying B. Why not A? This also grows on skew media. Skew पे तो दोनों grow करते हैं. Based on skew, I cannot say. Skew तो दोनों का है. Skew तो दोनों भाई भाई. The two brothers. You got my point. Why it is not A? Why it is not A? Because this is urease positive, this is urease negative. It is the urease test which is differentiating the two. Skew के basis पे नहीं बोला. Skew तो दोनों में है. Yes, urease word is important. So that's why correct answer is B. ठीक है. Coming on the next, Campylobacter is not important. It causes only diarrhea. ठीक है. Coming on the next, Pseudomonas. Come directly on the Pseudomonas. Can I come on the Pseudomonas? Is it given in your notes? Notes में है सब? I'm not following the notes. You are having everything in your notes? Can I continue with the Pseudomonas, the next organism? Pseudomonas, Haemophilus, Drusilla, Bordetella. 4 ही बचे हैं. 5 minutes, 5 minutes, 5 minutes, 20, 25 minutes we will pack. We will pack these. And we are left with Enterobacteriaceae and the miscellaneous. इतने बच जाएंगे, है ना? So it will take 1.5 hours tomorrow for finishing these 4 and the Enterobacteriaceae. So first, before breakfast, we will complete this. And after that, we will start with virology tomorrow. But let me finish these 4 small right now. ये छोटे-छोटे तो अभी कर लेते हैं. ठीक है? So can I start with the Pseudomonas? You have the notes, I guess. So let me continue with Pseudomonas. The next one is the Pseudomonas. Have I taught you we have only 2 pigment-producing bacteria in the morning? What are the 2 pigment-producing bacteria? एक तो मैं पढ़ा चुकी हूँ. One of them I already taught. So I already taught you Staphylococcus. It produces golden yellow pigment. Have I taught you in the morning? Oil paint appearance. This is the second bacteria which produces pigment. So Pseudomonas is a pigment producing bacteria, है ना? और क्या पता है? It's a pigment producing. तो मैं पढ़ा रही हूँ आपको Pseudomonas. I'm teaching you Pseudomonas. So first thing I taught you, it's a pigment producing bacteria. पहली बात, ठीक है. It produces 4 type of pigment, है ना? Most common is green. उसके अलावा blue भी है, yellow भी है, black भी है ना? Red भी है. So most common is green. ठीक है, चलो, है ना? What are obligate aerobe? Obligate aerobe कौन से हैं? Aerobe. So the mnemonic is nesty, pest, must breathe. का P क्या है? Pseudomonas. So you know it's the obligate aerobe. It's a pigment producing bacteria. Not only this, you know the mnemonic DPSS, Delhi Private Secondary School का P क्या है? Again Pseudomonas. देखो mnemonic पता है तो कितना कुछ. I have not started the chapter yet. मैंने chapter तो start ही नहीं किया है Pseudomonas. But I know this mnemonic, this is Pseudomonas. I know this mnemonic, this is Pseudomonas. And I know it's a pigment producer. तो कितनी चीजें तो मुझे पहले से पता है. So this is a way, है ना? Mnemonics याद किया तो आधी माइक्रो तो ऐसे ही हो जाती है. Okay, so read it now. Pseudomonas. Pseudomonas is the obligate aerobe. It's the pigment producing bacteria. I know it's the obligate aerobe because it is coming in nesty pest must breathe. है ना? So it is the obligate aerobe. It's the pigment producing bacteria. Only 2 bacteria produce pigments, Staph and Pseudomonas. I know that. है ना? It is the most common cause of hospital acquired infection known as nosocomial infection. You know what is nosocomial infection? The infection which occurs in the hospital. Imagine I'm going to hospital with some complaint. I'm going to hospital, I'm having some complaint. This is a hospital. You know, hospital is the most contaminated place in this world. The most contaminated. I'm going to the hospital with some disease. And I'm having a disease A. I'm having a disease X. And because of which I'm entering the hospital. So I got admitted. I got admitted in the hospital. Okay, in the hospital. After 48 hours. After 48 hours of admission, I'm having another disease Y, which was not present in me at the time of admission. So this new disease Y I acquired from the hospital. And I acquired. So this is hospital acquired infection or nosocomial infection. After 48 hours of admission, which was not present at the time of admission. Admission के time मुझे वो disease नहीं थी. After 48 hours, I developed a new disease. That is hospital acquired. So Pseudomonas is the most common cause of that. Hospitals में बहुत common होता है. ठीक है? And cystic fibrosis के patient में भी most common infection Pseudomonas ही कराता है. Pseudomonas causes the most common infection in cystic fibrosis. Here 2 culture media, nutrient agar, selective media. Nutrient agar पे याद करो, Staph aureus nutrient agar पे कौन सा कलर दे रहा था? Here Pseudomonas aeruginosa. Both are pigment producing. हाँ? ये nutrient agar पे कौन सा कलर दे रहा था? Say मैम oil paint golden yellow colonies. है ना? ये बनाएगा green color की colony. Can you see the green colony? On nutrient agar the complete green colony because of the pigment. It is known as metallic sheen. It is known as metallixene या iridescence. What it is known as? Metallixene या iridescence. So learn the two words. Staph aureus, Pseudomonas aeruginosa. Learn here golden yellow pigment, here green pigment. है ना? Learn the appearance. Say oil, paint, appearance. Learn iridescence. Iridescence. है ना? या फिर metallixene. One and the same. Metallixene या iridescence. So learn these terms. यहाँ पे oil paint था pigment की वजह से, यहाँ पे iridescence है. So it is iridescence. Iridescence या metallixene. That is on nutrient agar. Selective media citrimide. Citrimide यानी डेटॉल. हम बैक्टीरिया को डेटॉल से मारते हैं, ये तो डेटॉल पे ग्रो करता है. This bacteria grow on Dettol. It is very resistant, very difficult to kill. So selective media is very unique. Citrimide, very unique. Citrimide. Okay, okay. It is citrimide. Mechanism of action I already told you. DPSS. याद आया? DPSS का P है ये. It inhibit protein synthesis. So bacteria do not enter the cell. Toxin enter the cell. After entering, it inhibit elongation factor 2. No elongation factor, no ribosome, no protein synthesis. The cell will die. You already know that. Say yes. DPSS का P है ये. So tell me the diseases it causing. Most common it is causing hospital acquired infection. Hospital acquired में क्या क्या होगा? Hospital admit होने में क्या क्या infection होते हैं? 4 infections are most common nosocomial infection. 4 infections are most common nosocomial infection. Number 1, the person who is on ventilator. The person who is on ventilator, it is causing pneumonia in them. Ventilator associated pneumonia. है ना? Ventilator associated pneumonia. The person who is having catheter, it is causing UTI in them. So catheter associated UTI. है ना? Urinary tract infection. The person who is having central line, you know what is central line? उनमें sepsis हो जाता है. So central line associated bloodstream infection. ठीक है? And the person who is having surgery की dressing कर रहे हो आप, so surgical site infection. इन चारों का most common cause pseudomonas है. Don't learn बड़े बड़े नाम नहीं याद करना, डायग्राम में देख लो. So ventilator में, UTI में, catheter associated UTI, surgical site और central line. इन चारों का most common cause क्या है? Most common cause is pseudomonas. इन चारों का, ये चारों hospital में ही होते हैं. All these 4 infection occurs only in hospital, and the most common cause of all 4 is pseudomonas. You got it? It is pseudomonas. So this is most common cause of 4 type of hospital acquired infection. Say yes. Apart from that, it is the most common cause of skin infection. Many skin infections, है ना? With skin infection, for example, burns. You know burns में क्या होता है? Severe burns में ये सब rupture हो जाते हैं. Blisters are there, and the blisters get rupture. So patient can have infection in that blisters. Most common cause of infection in burn is pseudomonas. है ना? Burn में भी pseudomonas होता है. उसके अलावा it can cause ichthyoma gangriosom. उसके अलावा it can cause dermatitis. है ना? उसके अलावा it cause toe web infection. Can you see toe web infection? It can cause green nail. See, this is not nail paint. This disease is known as green nail syndrome. What it is known as? Green nail syndrome. Okay? Okay. No, Saeed, hospital will not be liable for that infection. You are a doctor now. Listen, what is the most common cause of hospital acquired infection or nosocomial infection? The most common cause is the doctor. Yes, you, me, we all are most common cause because we have many patients in our hospital. So what we people do, the doctors do, we examine one patient, है ना? We examine one patient and without sanitizing the hand, we touch the other. So what we have done, we have transmitted one patient infection to another. So this patient was already having an X infection. We have transmitted X to other. So it is a new infection for this patient. So the doctors are responsible for that. So sanitize hand, the hand hygiene before touching every patient, after touching every patient. That is the best way to prevent the nosocomial infection. Hospital do not take any liability. Okay, chalo. Can we go ahead? So this is green nail syndrome. Green nail syndrome. Okay, green nail syndrome. So these are the diseases caused by pseudomonas. We are done with pseudomonas. Chota sa bacteria is pseudomonas. Okay, pseudomonas. We have learned it's the pigment producing bacteria. Say yes. Okay, it's a strict aerobe. Say yes. It's the most common cause of hospital acquired infection or nosocomial infection. The mechanism of action, it inhibits protein synthesis by inhibiting elongation factor 2. So these are the main things. है ना? On nutrient agar, it give iridescence या metallixene because of the pigment. Read the question, tell me the answer. Quick, fast. Read the question, tell me the answer. An organism grow on the agar, show green colored colony. Very easy question. Of course, the answer is pseudomonas. Tell me the selective media. What is the selective media of pseudomonas? What is the cell activity of Pseudomonas? I told you na, citrimide agar. It's citrimide, very important. Citrimide agar, don't forget that. ठीक है? So coming on the next bacteria, Haemophilus, है ना? 3 more to go. Haemophilus. So the next bacteria is Haemophilus. Very easy. Haemophilus. Let's start Haemophilus. नाम में उसका मीनिंग है, split the name. Haemophilus. Hemo मतलब blood और philus मतलब loving. It's a blood loving bacteria. It loves blood. Without blood, it cannot grow. Why it cannot grow without blood? Because it requires 2 factors which are present in the blood. In the blood, factor 10 and factor 5 both are present. So let me draw blood vessel. In the blood vessel, let me draw the RBCs. These are the RBCs. है ना? Factor 10 is present everywhere. Factor 10. And factor 5 is present intracellular. Please learn. Factor 10 is present everywhere, free in the blood. Factor 10 is free. And factor 5 is intra-RBC. वो RBC के अंदर होता है ना? Now I am teaching you Haemophilus. What I am teaching you? Haemophilus. How many type of Haemophilus in your syllabus? There are many Haemophilus. Some require only 5. Some require only 10. Some require both. ठीक है? Based on which we are classifying Haemophilus. So let's classify Haemophilus. I guess this is given in your notes. थोड़ा आगे पीछे होगा लेकिन होगा. Haemophilus में थोड़ा आगे दिया होगा ये. But it is given in your notes, है ना? So let's classify Haemophilus. Listen, everyone on the screen. Let's classify Haemophilus. There are some Haemophilus which require only 10, not 5. There are some Haemophilus which require 5, but not 10. And there are some Haemophilus which require both. Okay, so how to classify? How to learn? Listen, the one which require only 10 is Ducri and Aerophilus. They require only 10. The one which require only 5 is Haemophilus para-influenzae, Haemophilus para-hemolyticus, and Haemophilus para-aphrophilus. ठीक है? These require only 5, not 10. The one which require both is influenzae, the main hero of this chapter. Haemophilus influenzae, Haemophilus aegypticus, and hemolyticus. Now I know what you all, है ना? You all are scratching your hand कि कैसे याद करना है? ये क्या गंदा सा मैम बता रही है ना? मुझे पता है तुम लोग ऐसे ही सोच रहे हो. Got it? Let me tell you a way. I will simplify it. I will give you a mnemonic. So tell me the Haemophilus which require only 10, not 5. Tell me a Haemophilus which require only 5, not 10. Tell me the Haemophilus which require both 10 and 5. ठीक है? I will give you a mnemonic. Don't worry. हर चीज का mnemonic है हमारे पास. ठीक है? चलो, listen. कैसे याद करेंगे? 10 को हिंदी में क्या बोलते हैं? 10 का स्पेलिंग कहां से स्टार्ट होता है? D for 10, D for Ducri. Ducri require only 10. You will not forget in your life now. Ducri require only 10. Because D is Ducri, D is 10. है ना? 10 का 10. D Ducri, D 10. Say yes. है ना? Okay. So what is 5? 5 को हिंदी में क्या बोलते हैं? 5 का स्पेलिंग स्टार्ट विद P. All para start from P. Yes, all para. Para influenzae, है ना? Para Haemophilus, para Aphrophilus, para. P मतलब 5, P मतलब para. Say yes. And the one which require both, है ना? The one require both. Both किसको लगता है? उसके लिए एक निमोनिक है high, है ना? HAI, high. So what is high? Say the full form. Haemophilus hemolyticus, Haemophilus aegypticus, and Haemophilus influenzae. I'm trying hard, है ना? समझ में आ रही है क्या बात? है ना? I'm trying very hard to learn you. ठीक है? So please, you get many questions on that. समझ में आई बात? You want to revise again? Tell me the Haemophilus which require only 10, not 5. Tell me the one which require 5, not 10. And tell me the one which require both. Who will tell me? 10 का बताओ. Haemophilus Ducri, D for 10. यहाँ पे Haemophilus all para. कोई सा भी पैरा ले लो, है ना? एंड यहाँ पे Haemophilus high, HAI, है ना? Haemophilus hemolyticus, Haemophilus aegypticus, Haemophilus influenzae. Okay जी. So this is only 10, this is only 5, this is both. Okay, है ना? So this is how you come to know. Just suppose I'm having a specimen of the Haemophilus with me. This is my specimen of Haemophilus. I know this is Haemophilus, but I want to know which one. Is it Ducri? Is it para? Is it influenzae? कैसे? So what I will do, I will take a nutrient agar. I will take a blood agar. I will take a blood agar. On this blood agar, I will take 3 filter paper. है ना? This is filter paper dipped in 5. This is filter paper dipped in 10. And this is filter paper dipped in both. 10 plus 5. है ना? And I will try to inoculate. Now I will try to inoculate this specimen here. I will do the inoculation. Now if I get a growth only here, not on 5, not on 10. So it means it is high. It is one of the high. अब high में से तीनों कौन सा है? I don't know. लेकिन it requires both. That's why it is growing only here. or else there is a possibility it is growing at 5 also and here also. So it requires only 5. So it is one of the para. है ना? Or else there is a possibility it grows at 10 also and here also. So it is Dukri. How many of you got it? How many of you got it? You got the 3 possibilities? Say yes or no. Shall I say again or you got it? So this is known as disc test. You can see. Can you see in this disc you can get beautiful image based question. इस image में देख के बताओ where you are getting the growth. In this image where you are getting the growth. You are getting the growth along the 5? Say no. Along the 10? Say no. Along both. Along both. We are getting the growth along both. See, यहाँ पे growth है. So which one? One of the high. I can't say अब कौन सा है, लेकिन high है. I can say it's not Dukri. I can say it's not para. So you can get beautiful image-based question on this concept. हाँ बोलो, सब हाँ बोलो, है ना? ठीक है. So this is the classification. So we started Haemophilus and we have seen the classification. So here I am going to teach you 3 Haemophilus. The most important is Haemophilus influenzae, the hero, and one line on Dukri. Dukri और एक और है कोई, I guess one more Aegypticus. So one line here, but main is Haemophilus influenzae. I will teach you a little bit detail. ठीक है? एक चीज याद है, influenza do not cause influenza. It's a misnomer. Influenza नहीं कराता. Initially we thought influenza causes influenza. That's why we named it Haemophilus influenzae. Later on, the discovery was that influenza is caused by a virus, influenza virus. We have influenza virus now, orthomyxo. I will teach you tomorrow. That causes influenza, but we have not changed its name. So Haemophilus influenzae do not cause influenza. Remember that. Remember that. So let's start influenzae. What is the culture media now? You tell me Haemophilus influenzae require what? You tell me. Haemophilus influenzae require what? It requires 5 or 10 or both. I'm giving you 3 options. जल्दी बताओ, 5 or 10 or both. जल्दी बताओ. जल्दी बताओ. You tell me. It requires 5 or 10 or both. अरे say people, it requires both. High का I है, ये HAI का I. So it requires both. It requires 10 also, it requires 5 also. है ना? Now I'm asking you 3 questions. Will it grow better on blood agar or chocolate agar, and why? Will it grow better on blood agar or chocolate agar or both? Both? Nitin is saying both? Acha, both. It requires both. No. You tell me whether it will grow on blood agar or whether it will grow on chocolate agar or whether it will grow both equal. Why? Lalit is saying blood agar. Reena is also saying blood. Elizabeth is also saying blood. Prabingal is saying chocolate. Why Prabingal? Why you are going opposite of your family? Why chocolate? Okay, Devbrabat is also saying chocolate. Okay. Why Kartika? Why blood? You should tell the reason also. Chalo. Answer is chocolate. It will not grow on blood, it will grow on chocolate. Chocolate bhi to bloody hai, heated blood. Chalo, let me tell you the reasons. Listen, everyone on the screen. Dekho, let me draw the blood agar, let me draw the chocolate agar. This is blood agar, this is chocolate agar. Blood agar, the RBCs are intact, like this. RBCs are intact. So, factor 10 is available, it's free, but factor 5 is not available, it's intracellular. The factor 5 is intra-RBC, it's not available. So if you try to grow Haemophilus influenzae here, it will not grow. It will not grow because it requires both. 10 is available, halaki. Although 10 is available, but 5 is not available, so it will not grow. No growth or scanty growth. No growth or scanty growth. But what is chocolate agar? Heated blood agar. Once you heat the blood agar, the RBC will get rupture. All the RBC will get rupture on heating. So 10 was already available. There was no problem in 10, but 5 also become free. So 5 and 10 both are free now. Now try growing Haemophilus influenzae. You will get abundant colony, bhar ke, abundant growth. This is the reason. Say yes now. You will never forget now. So there is no growth on blood agar. Learn the summary. There is no growth on blood agar, and there is abundant growth on chocolate agar. Learn it with reason. Why there is no growth on blood agar? You should be able to tell the reason. Man, 10 is available, but 5 is not available. So this is the reason. And why abundant growth on chocolate agar? Because on heating RBC rupture. So 10 and 5 both are free. ठीक है? This is the reason. ये दोनों पे PYQ है. No growth on blood, abundant on chocolate. You should know the reason also. Everyone got it? ठीक है? समझ में आई? लेकिन मेरी तो जिद है कि मुझे तो ब्लड पे ही ग्रो करना है और अच्छा ग्रो करना है. करो कोई तरीका निकालो. I want to grow on blood agar. I want to grow only on blood agar. I do not want to grow chocolate. Haemophilus influenzae. And I want a good growth. Is there a way? I'm having a wish. Do something now for me. Please. Do something. ठीक है? There is a way. Let me tell you a way. Let's take a streak of Staph aureus. You know the first bacteria I taught you? Staph aureus. You may be thinking, ma'am, Staph aureus का यहाँ पे क्या लेना और क्या देना? Why you are taking Staph aureus? There is a property. Staph aureus secrete. It synthesize and secrete factor 5. It synthesize and secrete factor 5. Huh? No, I'm not adding Uday factor 5. I'm adding a streak of Staph aureus. Staph aureus will synthesize and add factor 5 in that. 10 was not a problem. 10 is available everywhere, and now 5 is also available. Now try growing. Now, in this manner, you try growing Haemophilus influenzae. Will you be able to grow? This is blood agar only. This is blood agar. On the blood agar, I have taken a streak of Staph aureus at the center. Now I am trying to growing. So near the streak, the colonies are bigger. And as we move away from the streak, the colony size goes smaller, smaller, smaller. Why the colony are bigger near? And as we are moving away, the colony becomes smaller, smaller, because maximum concentration of factor 5 is available near the streak. As you move away from the streak, the concentration decreases, so colony size decreases. Can you see the appearance? It is looking small, small satellites. Small satellites are there. This is known as satellitism. Satellitism. Very important question. Very important question. So what happened on blood agar, you tell me? What happened on chocolate agar, you tell me? And what happened on blood agar with Staph aureus streak? You write down the answer. Blood agar, no growth or scanty growth. Chocolate agar, abundant growth. And blood agar with Staph aureus, it is satellitism. है ना? See blood agar. See the diagram. See blood agar. Say no growth. Hardly small colonies are there. है ना? Scanty growth. Because 10 is available, but 5 is not available. It's intracellular. है ना? On chocolate agar, see abundant growth. Big colonies are there. Big. Because RBCs get ruptured. So 10 was already there, but 5 also released. But blood agar with Staph aureus ka streak. है ना? This is known as satellitism. How many of you got it? Satellitism. Now on this image, let me ask you 4 questions. Image 8, question anek. 4 questions on one image. First, tell me the streak is of which bacteria? The streak. Name the bacteria, which is the streak. Name the bacteria. Your answer will be, ma'am, Staph aureus. I'm not waiting for the answers. है ना? It is Staph aureus. Then I will ask the colonies of which bacteria. ये colonies जो हैं वो किस bacteria की? Streak नहीं, colonies. You will say, ma'am, Haemophilus influenzae. The second question. The third question, why we are adding Staph aureus? The answer is, it is providing factor 5. है ना? The fourth, what is this appearance known as? You will say satellitism. है ना? So 4 questions on one image. This is satellitism. Say yes. Haemophilus influenzae causes many disease but not influenza. It do not cause influenza. It causes meningitis. Okay. It causes epiglottitis. And it causes pneumonia. है ना? But it do not cause influenza. It causes meningitis, epiglottitis, pneumonia. But it do not cause this thing, influenza. So main hero is done. Haemophilus influenzae is done. Now one side hero Haemophilus ducrei कर लेते हैं and Haemophilus is done. So main hero तो हो गया. It requires both 10 and 5. है ना? What about ducrei? Ducrei का क्या है? It requires only 10, no 5. It requires only 10. D for D. ठीक है? So D for ducrei. ठीक है? D for ducrei. ठीक है? So ducrei is showing bipolar staining. Have I taught you bipolar staining? याद आया क्या? Safety pin. Safety pin कौन present था कल? Safety pin appearance. 5 bacteria show safety pin appearance, can produce very high yield. Yield ka, I'm sorry. H is Haemophilus ducrei. H is Haemophilus ducrei. So number 1, it shows bipolar staining. Number 2, in the blood. Just suppose it is present in some human blood. So it is having 2 appearance. Number 1, railroad. It is like the rail ki patri. English में क्या कहते हैं? Railroad appearance. They travel in parallel line. It is known as railroad appearance. Number 1, in the blood. Or else, they will make a group like the fishes. It is known as school of fish. It is not fish in stream. देखो अंतर है. अगर क्वेश्चन में आए fish in stream, तो what is the answer? और एग्जाम में आए school of fish. Meaning वही है, the group of the fishes. But the wordings are different. This one is Vibrio cholerae. This one is Haemophilus ducrei. Please learn it. Okay? So these are the 2 appearances you have to learn. Clinical feature, Haemophilus ducrei causes a STD. The Haemophilus ducrei causes a sexually transmitted disease. It causes an STD. The name of the STD is chancroid, soft sore. It causes a disease, chancroid. So on the genitals, you can see the penis. On the penis of a male, you can see the genital ulcers. These are bleeding. These are soft. है ना? These are bleeding, soft, painful, tender, but non-indurated. ठीक है? That is chancroid or soft sore. So read the question. Tell me the answer. We are done with Haemophilus. Haemophilus भी हो गया. Read the question. Factor 5 and 10 both are required for more. For which? The factor 5 and 10 both are required for high. High में से कौन आ रहा है यहाँ? Yes, high का I. It's influenzae. ठीक है. If I change the question, only 10 is required. For what? For which of it? Only 10 is required. Only 10. So what is the answer? In that case, your answer will become B, Dukri. And if I say only 5, the answer will become both the para. So you can understand. Okay? So you get many questions on that. Read the next question. Read the next question. Hemophilus para-influenzae requires which factor? Only 10, only 5, both or none. Hemophilus para. What is para? Para is punch. So P for para, P for punch. It requires only 5. Only 5. So in this way, you get many questions on that. Okay, Ji. So we are done with all these bacteria till now. These are done, these are done, these are done. Only 2 are more. So let's finish it in the 5 minutes. उसके बाद फिर सिर्फ मिसलिनियस बचेंगे. चलो, Brucella is again a zoonotic disease. It's again a zoonotic disease. I taught you one more zoonotic disease, Bacillus anthrax. Brucella is also a zoonotic disease. It comes from the animals. It comes. So there are 4 types of Brucella. Brucella melitensis, abortus, Swiss, or canis. Melitensis come from the sheep and goat, camel. Abortus come from the cattle, buffalo. Swiss come from the pig, and canis come from the dog. Don't learn it. It is coming from the animals. Okay, just learn that. It's a zoonotic disease. Learn the selective media. The selective media is Castanenda, biphasic brain-heart infusion. Why it is known as biphasic? Some medias are solid, some are liquid. It is double. You can see the agar slope also, and you can see the broth also. It is double. It is solid also, it is liquid also. That's why it is biphasic. Castanenda biphasic, and it is also capnophilic. मैंने बोला ना capnophilic 2 ही होते हैं, Legionella and Brucella. LB, BL, कुछ निमोनिक बना लो. The only 2 bacteria which are capnophilic, they require carbon dioxide. मैं बता चुकी हूँ, है ना? It causes a disease, है ना? It is known as, it is having a typical triad. The name of the disease is undulating fever. Fever आता है, fever come, and without any treatment it goes away. Then again come, then again go away. Then again it is known as undulating fever. It is known as undulating fever, also known as Malta fever. या फिर Mediterranean fever, because it was first time discovered in Mediterranean, Malta island of the Mediterranean. Malta island पे discover हुआ था. That's why undulating fever, Malta fever, Mediterranean fever, number 1. Number 2, arthritis, bone pain, and number 3, hepatosplenomegaly, the triad of Brucella. The triad of Brucella. Please learn the 3 things. है ना? कुछ ज़्यादा question नहीं आते हैं Brucella में से. So Malta fever is caused by Brucella. Nothing important. And last one is Bordetella. It will cause whooping cough. है ना? Bordetella क्या appearance याद करने हैं. The 3 appearance of the Bordetella. There are 3 appearance in the Bordetella. The first appearance in the Bordetella is thumbprint appearance. है ना? कभी अंगूठा लगाया है अपना कहीं पे? So see, it is looking like that. Can you see here? Small Bordetella. They form the loose clump. It is looking like thumbprint appearance. Please learn this appearance, Bordetella. Bordetella pertussis. Number 2, culture. Learn the selective media. The selective media is Regan-Lowe media. Regan-Lowe या Borded Gengu glycerin potato agar. So you have to learn the selective media. Selective media तो सबके learn करने ही पड़ेंगे. On selective media, there is typical appearance of the colony. Can you see the colonies? Colonies कैसी लग रही हैं? The colonies are looking like what? They are looking like pearls. मोतियों के जैसी हैं. These are known as bisected pearls. Bisected pearls colonies. Or they are looking like mercury drop. They are looking like mercury drop, है ना? Mercury drop or bisected pearl. So learn the 3 appearance: thumbprint, है ना? Mercury drop and bisected pearl. Learn the 3 appearance. Mechanism of action I will not tell. You tell. I told you 10 bacteria ना, ये कहाँ आता है? Bordetella कहाँ आता है? Does it come? I told you the 4 bacteria which inhibit protein synthesis, उसमें आता है. Or I told you 4 bacteria which increase cyclic AMP, उसमें आता है. Or I told you 1 bacteria which inhibit GABA glycine, उसमें आता है. Or I told you 1 bacteria which inhibit acetylcholine, कहाँ आता है? Yes, Bordetella comes here. Be very efficient boy. One is Bacillus anthrax, one is Bordetella. It increases cyclic AMP. So it increases cyclic AMP in the lung, है ना? So don't. That's why I told you all 10 together, एक बार में. So बार बार नहीं बताना. This is the 10वीं बार, 10th time I'm telling you this mnemonic. This is the last bacteria. ठीक है, चलो. So it is increasing cyclic AMP in the lung and causes a disease known as whooping cough. Whooping cough, pertussis or 100-day cough. It causes a disease, whooping cough, pertussis or 100-day cough. There are 3 phases: catarrhal phase, paroxysmal phase, convulsant phase. देखो, catarrhal phase में there is a running nose. पूरा टाइम नाक बहती है, running nose. It is known as coryza. It is highly infective. Catarrhal phase. It is transmitting the infection to multiple people at a time. Highly infective stage, है ना? Most infective. Second is paroxysmal stage in which patient cough. cough. This is a typical cough, not normal cough. It is whooping cough. What do you mean by whoop? So what is a cough, by the way? Cough is an expiration. It's like that. Forced expiration. है ना? So patient cough for 5 to 10 times non-stop. It's like that. So the glottis become closed permanent, very long. 5 to 10 times the glottis become closed. Now after 5 to 10 times, when the patient start inhalation, after when the first inhalation, have you seen whenever a closed pipe, a very narrow pipe, when the air pass from a narrow pipe, there is a sound. The sound is zzz, ऐसा करके sound आता है. So it's like that. So whenever the patient is coughing for 5 to 10 times, and whenever the patient is inspiring, it's like that. So after 5 to 10 times, it's like that. So that is known as whoop. Whoop is inspiration. After 5 to 10 times of the cough, the inspiration is the whoop. So that is known as whooping cough. 5 to 10 times of repeated expiration के बाद जब inspiration होगा, it is known as whoop. Yes, it is barking cough. So this is a video. Patient is coughing and then whooping. Typical sound is there. And convalescent stage में recovery antibodies. We are done. चलो. Tell me the most infective stage. तीनों में से most infective कौन सी है? Catarrhal. Most infective is catarrhal. और इसी को change कर दूं तो whooping cough किस में होता है? Whooping cough कौन सी stage में होता है? It's paroxysmal. और इसी को change कर दूं तो recovery कौन से phase में होती है? Convalescent. तो तीनों phase का 111 highlight you have to learn. We are done. We are done. So I would like to stop here. We have completed gram-positive cocci, gram-negative cocci, gram-positive bacilli. And among the gram-negative bacilli, except Enterobacteriaceae, we have completed everything. So technically, we are left with Enterobacteriaceae. From here, only one bacteria, but it's big, and 4 small miscellaneous. These are also very small. I wish I could have time कि आधे घंटे में हो जाते, but ठीक है ना? So these 4. So this is the pending. Okay जी, give me a thumbs up. मजा आया बैक्टीरियो पढ़ के? हाँ, इतना भी आज हो जाता अगर 1 घंटा और होता मेरे पास तो एक ही शॉट में होता. So I'm sorry I couldn't complete it in one shot, but I will start tomorrow. So tomorrow timing, it's not 9, maybe I guess it's 9:20. It's 9:20, है ना? I have to drop school to my baby. 2 दिन तो छुट्टी थी उसकी, है ना? लेकिन after dropping school, after 9:20, we can start our marathon till 7 or 8 in the evening PM. Whatever, 7 or 8, we will be able to complete it. Tomorrow's plan, first I will finish the bacterio, okay? I will finish the bacterio, whatever pending, and then we will start viro or parasito, whatever you say. One thing we will complete tomorrow, either viro or parasito. So maybe we will complete viro tomorrow. So bacterio plus viro, tomorrow's plan. Okay, so tomorrow the timing is at 9:20. I will confirm with the team. So please confirm the timing for tomorrow from the team. I guess it will be 9:20, but I will confirm with them. Thank you. Bye-bye. Be on time. Okay, ji. Bye-bye. Chalo, I'm ending the class.